at the last chapter region around the world. Nick, do you want to start us off?
Indeed. Thank you very much, Christine, and thank you so much to those who've gone before. Um, for those just joining from Europe who haven't seen yesterday afternoon or the tremendous amount of talks done overnight European time, we've had a wonderful sessions starting at about midday or 1 p.m. European time yesterday, starting with the North American region, going through all sorts of pediatric and adult talks, followed by Latin America in Spanish and Portuguese for much of it. Then we've had Asia Pacific and most recently Southwest Asia and Africa chapter, with a great deal of talks on, particularly on some unusual topics for us with some unusual infections, etc. It's now EuroElso's turn. So thank you very much to the panelists and our speakers who are going to be joining us for the next 4.5, 5 hours. The sessions will be roughly 40, 50 minutes with some time for 15 minutes or so for discussion afterwards. So please do put any questions into into the chat, into the Q&A, or and we can chaperone those and discuss them with the speakers. I just wanted to start though with a few comments around the chapter. Clearly, this particular Congress is in honor of Bob Bartlett, who you will all know was the father of ECMO. He did a huge amount of basic research, a huge amount of clinical support, clinical research, and was tremendously supportive of the development of ECMO globally. In addition to his boundless academic enthusiasm and the interplay between technology and clinical practice, he was also key to the development of ELSO and started the ELSO organization off. He was also key to the development of the chapters. He recognized that there was a need for sharing information, for sharing leadership, and for growing ECMO, which has happened with the development of the chapters. EuroElso was the first of those, and there was a meeting in one of Alain Coms's congresses in Paris, which kicked off EuroElso. And Bob was fully supportive of this from the start, and was fully supportive of the growth that we've seen in EuroElso over the last nearly 15, 16 years now. He's attended most of our congresses until recently and has been, I know, an inspiration to many of you. He also directly influenced a lot of the clinical progress of ECMO in Europe and around the world. And if we look particularly to some of the big studies that he's influenced, the UK collaborative trial for neonates, he was very supportive of and provided a lot of some of the background unpublished data to help support the development of that. He was tremendously helpful for Giles Peake and Richard Furman with CSER, and of course helped to leverage the experience that occurred in Australia with the development of H1N1 back in 2010 into understanding how that, in combination with the CSER study, could be used for respiratory ECMO. And I think you'll all agree that we've seen that tremendous change in Europe with the rise of respiratory ECMO. More recently, of course, we're seeing the rise of cardiac ECMO and most recently, huge amounts of work on ECPR, particularly from Jan Błachowicz. I'd like to thank those our co-chairs who've come before me. So Peter Alexander for the US, John Fraser Rodrigo Diaz, Ibrahim Fauzi, and of course, hiding in the background, Alex, Lauren, Christine, Peter, to keep the tech running and to keep everything moving smoothly. And for this session, we've got some great talks coming up. We've decided to focus on some of the enormous changes that we're seeing in particularly the technology for the next few hours. So we're going to be looking at a range of talks over the course of the EuroELSO session, from VADS to transport to material science to monitoring to pharmacokinetics, ECPR, of course, and rehabilitation. And we'll aim to bring everything to a close around 12:30 or so Central European time. We are, of course, flicking back and forth across a few time zones, which is causing me personal havoc, but that's what I get for living in the UK. We've got joining me this morning, Jutta Arens and Sylvia Mariani. Sylvia and Jutta, maybe if you could introduce yourselves, please.
Yes, sure. Thank you for the introduction, Niklas. My name is Jutta Ahrens. I'm a mechanical engineer and working as a professor at in engineering organ support technologies at the University of Twente. And allow me two sentences more about Bob, because what he also did is that he brought the engineers into the game. And he was very fond of bringing clinicians and engineers together. And half of my today's collaborators in projects I was introduced to by Dr. Bartlett. So I'm really, really grateful for that opportunity. And I think that's an important part of EuroELSA as well, that we also have the engineers, the perfusionists, and everyone on one table. So good morning, everyone, and enjoy the session.
Yeah, good morning, everybody. My name is Silvia Mariani. I'm a cardiac surgeon from Italy, and I'm really happy to be here side by side with engineers, as you just said, intensivists, and this shows really the multidisciplinary approach that we have in this field. And, being here is a great honor for me. As I was growing up as a surgeon, I always look up at Bob Bartlett as an example and for his humanity first. And I will remember forever when he told me that the most important thing was the last page of the red book, the picture where he was hugging a patient. And that's a sentence that's sticky in my mind and still guides me every day in my daily work. So I'm really, really grateful for being here today, to Bob Bartlett and to all the organizers and colleagues who are contributing to this event and to the everyday's work in a clinic for our patients. So I'm really looking forward to all the talks today.
Lovely. Thank you both. Well, it falls to me to kick things off. So I'm going to introduce Dr. Thomas Schlogelhofer, who is the technical director of the MCS program at the Medical University of Vienna and works in the Department of Cardiac and Thoracic Aortic Surgery at the Center for Medical Physics and Biomedical Engineering in Vienna. We're going to start with a very, very common and practical problem, which is Thomas is talking to us about how to keep the cannulae in place and look at the latest in ECLS cannula fixation for prevention of dislodgement. Thomas.
So, Nicholas, thank you very much for the introduction. I would like to thank all the organizers for giving me the opportunity to present here in this very important symposium. I'm going to share my slides with you. As already said by Nicholas, my talk will be about staying in place ECLS cannula fixation for dislodgement prevention. These are my disclosures. I think, as we all know, that also recommends a secure fixation of ECMO cannulas, and this is basically at a minimum of 2 points to prevent dislodgement. Common methods, as we all know, are sutures or securement devices, so-called adhesive anchors, and they are often combined with proper dressings. The key goal is, as we all know, stability to avoid life-threatening issues like accidental decannulation or even preventing movement during patient positioning. As you can see on the right-hand side, why? Because this, as we all know, could lead to impaired blood flow, infection, mechanical circuit malfunction, air embolism, or major bleeding. And what we also see in the ELSA recommendations is there are, first of all, 2 points minimum securement. So secure each cannula, ECMO cannula at least twice along its length. And of course, it depends dramatically where and which cannulas we're using. For example, for femoral cannulas, secure them above the knee to counter movement from the knee flexion. If we look to a recent survey by Ball and colleagues, we can see ECMO cannula securement is a small detail with big consequences. There are obviously around the globe several approaches how we can secure cannulas. And in this survey of 45 international centers, we can see that 34% of the respondents reported cases where the cannula had a malposition, dislodgement, or accidental decannulation within the previous 5 years. And of course, this was leading to adverse patient outcomes. Among those incidents, it's really impressive that more than 50% of the centers provided detailed accounts that this was based due to inadequate cannula securement as the leading cause. And additionally, 3 or 4 of the survey participants indicated that the development of an international evidence-based guideline for ECMO line management would be really instrumental in enhancing bedside practices. So if we look to ECMO cannula securement versus dislodgement, I think we should say that most discussions frame cannula fixation quite binary. Either the cannula is in or out. But mechanically, we can see that that's not how failure happens. Obviously, fixation of cannulas exists on a spectrum, and I tried here to summarize this, that specifically we're talking in the routine care during patient movement, the so-called micromotion, which is up to 5 millimeters, more on this in the next slide as well. There is, of course, progressive movement where fixation has more stress and is even loosening. And the main problem, of course, the clinical dislodgement, where we, of course, can see then bleeding and an intervention is needed. So why is micromotion as a clinical concept really mattering? What do we know about it? There are some key clinical observations that even these small movements were associated, especially with local complications and infection risk. And in a recent publication of Lucchini and colleagues, they identified these so-called micromotions during prone positioning, suggested managing displacement to 5 millimeters and really focus on infection prevention than mechanics. So the important limitations of this clinical study was it was a very small cohort. It was specific, as I said before, to prone positioning. And of course, there were no mechanical force or stiffness measurements performed, as this was a clinical study. So what are the major knowledge gaps? So the main questions for us were how much force causes really a movement? How do fixation behaves at different displacement ranges, and how consistent are various methods to fix cannulas. So the aim of our study, what was recently published, was as we see repeated small movements during routine care of our ECMO patients, and there are obviously different mechanical and different fixation methods in the micromotion, which is still unknown, the mechanical behavior. And as I said before, the prior work focused really mainly on the clinical context, no mechanical performance. Our study rationale was to isolate and compare the mechanical performance of ECMO cannula fixation methods under controlled displacement conditions, with emphasis especially on micromotion. So you can see basically the material and methods the experimental setup on the right-hand side. So we performed basically measurements with a tissue model, porcine skin muscle sections, which we took from the slaughterhouse. We inserted cannulas, 21 French venous ECMO cannulas, biomedicals, inserted them using a so-called modified Seldinger technique, and inserted them to the 45 centimeter mark. So then, as you can also see, we removed the hair. Tissue was mounted in a custom-made holder, what you can also see, as I said, on the right-hand side. We then did a one-to-one randomization, started, for example, once with the sutures and one with sutures plus adhesives. And basically, the comparison was to compare standard free silk sutures. You can see this also in graph A here, compared to one silk directly suture at the cannula tip and adhesive anchoring devices. And for the adhesive anchoring devices, we compared basically the calf grip, as you can see it on the right-hand side in graph B, and also the Hollister fixation, which is a quite commonly used anchoring device. The mechanical testing included basically an uniaxial universal testing machine. We used then a load cell, which you can see also here, 1 kilonewton, and performed basically a pull test with upward displacement up to 50 millimeters with 250 millimeters per minute. And as main outcomes, we measured the load, especially in the range of 15, 25, and 50 millimeters. I would like to address a little bit more the mechanical characteristics and metrics, what we measured. So as I said, the force already at the displacement to remove the cannula was measured. As I said before, very important, below 12 millimeters is definitely the micromotion range. The stiffness was also assessed, so the resistance to movement over a given displacement range. And we also calculated the so-called dislodgement energy, so the total work which is required to displace a cannula from the optimal position. So just to summarize here, material and methods, traditional sutures, I think not a big surprise for you. They are widely used, familiar technique. The cost, of course, is a pro. Cons, it's a point fixation, as we all know, risk of skin tearing, rupture, or cannula damage. And on the other hand, these adhesive anchoring devices, they are available in variable sizes. They have even for skin latex-free hydrocolloid surfaces, distribute, of course, the load, flexible fixation. They have no skin penetration, can be worn up to 7 days, both of them. The cons, there is less historical data. Application technique matters, of course, and cost. They are, of course, a little bit more cost intense than sutures. Let's have a look to the results briefly. You can see here the measurements on the left-hand side of the sutures, and on the right-hand side, the dislodgement forces for the adhesive in this term here for the calf grip device. So overall, we performed 26 measurements with anchoring and also with sutures. And I think what we can nicely see already here in the red line is, although this was performed, all the sutures by one single surgeon in a standardized way, there is a greater variability at the maximum force dislocation. So obviously, even as I said, one surgeon did it, there is big variation in the sutures and the forces. We can also see that some of the sutures, you can also see this in the graph here on the top left, that 27% of the sutures ruptured actually already at the maximum dislodgement at 50 millimeters, whereas the calf grip showed actually a greater flexibility compared to the sutures. Also, if we compare this in the free force areas, and especially, as I said before, I would like to address here the micromotion range. We can see that in blue, the adhesive had a higher force, so a higher load was needed to pull out the cannula, which is a good thing, obviously, compared to free sutures. This was comparable in the higher range, but especially in this micromotion range, the adhesive performed better than the sutures. As I also said, we had a look to energy and stiffness, and I would like again to address here in the 0 to 5 millimeter range, the calf grip adhesive showed a higher stiffness compared to the sutures and also a higher dislodgement energy. And this is indicating actually greater resistance to deformation in this initial range and potential beneficial compared to adhesives. Sorry. Then what about alternative methods? Because we were not only interested in this one adhesive, we also had a look to the really classical Hollister anchoring device. And you can already see here in B a problem, what we experienced in several of our tests, that this is really loosening, and comparing the Hollister adhesive with the cash grip, we saw in the micromotion area no differences, but at higher forces, obviously dislodgements, we can see that the cash grip outperformed basically the Hollister. So it can be recommended to use actually the cash grip and not the Hollister device as an adhesive. So to summary, and also here a conclusion, ECMO cannula fixation is not binary. It's displacement dependent. There are differences between fixation methods, and they are most pronounced in the early micromotion range. In this range, adhesive anchoring via the cash grip demonstrated greater flexibility, as we have seen here, and stiffness, which increased resistance to small movements. Mechanistically, I think reducing micromotion may help to limit skin stress, cannula constriction, local tissue irritation, and this could also support on the long-term patient comfort, early mobilization, although clinical validation is of course required. And this brings me also at the end of the day here briefly to the limitations of our study. It was of course only an in vitro model controlled laboratory setup, so it does not fully replicate the clinical environment, that's clear. Porcine skin model, no long-term wear or skin changes could be assessed, also like sweating, for example, which is definitely to be considered in a patient. The scope of testing was short-term, single direction load, no patient movement simulations so far. So what are the future directions for us as a community? I think we will need clinical correlations, so prospective clinical studies where we correlate also different anchoring approaches and correlate displacement with clinical outcomes, very needed. Real-world motion testing, so multi-directional loading and simulating also routine care and mobilization. And of course, it's also very relevant. What about long-term fixation behavior? So time-dependent mechanics and skin device interactions should be assessed in the future. So this brings me to the end of my presentation. Thank you for your attention, and I'm looking forward to the discussion later on. Thank you.
Sorry, we directly go to our next speaker, to Markus Granegger. Thank you very much, Thomas. Markus Granegger is Associate Professor of Cardiovascular Engineering at the Medical University of Vienna, and his research mainly focuses on artificial hearts, ventricular assist devices, and translational cardiovascular technologies. So he works at the interface of engineering and medicine to devices into the hospital. Markus, the stage is yours.
Perfect. Thank you so much for the introduction. It's a great honor to be here at the Bartlett 24-hour summit. I was asked to give an update on a novel ventricular assist device for pediatric patients. So I hope it's working. Yeah, as you know, cardiac insufficiency is a global challenge. I think I don't have to go into into more detail here. The theoretical solution for cardiac insufficiency in many patients is cardiac transplant. But however, as you know, we have a donor shortage, and that's why mechanical circulatory support evolved. And specifically, left ventricular assist devices are a standard therapy for durable heart support. Whereas in adult patients, the technology evolved towards intracorporeal rotary blood pumps, which are fully implanted with just a cable exiting the patient. In pediatric patients, the technology lags behind. So the only available pump for infants and small patients is the paracorporeal Berlin Heart X-Core device. So paracorporeal means it's outside of the body, and it's connected to a huge pneumatic driver. With this system, in contrast to the adult systems, there is no discharge from the hospital possible. And as these patients wait for a donor organ, sometimes more than a year, you can imagine what it means for the patient itself, for the development of the patient, but also for the families who sometimes need to move to a center close to a center who's able to provide this technology. We definitely have an unmet medical need for an intracorporeal pediatric system, but why are no pediatric systems available? One, of course, is the comparably low patient number and the very heterogeneous patient population. But on the other hand, we also have a very complex interaction between the patient and the pump, because interestingly, as you know, in pediatric patients, we have similar pressure conditions as in the adult population, but we have a lower flow. And interestingly, it was found that the lower flow is actually a challenge for these rotodynamic blood pumps. So we need to come up with an intracorporeal pediatric LVAD, which is small in size and has an excellent hemocompatibility at low flow conditions of approximately 1.5 liters per minute to treat also the smallest patients with an intracorporeal pump device. And so our aim was to develop such a system, and we came up with a miniaturized two-stage blood pump. I will come back to that a bit, why we chose this two-stage design, which fulfills the specific requirements for intracorporeal LVADs for small pediatric patients. So why did we select the multi-stage pump concept? So if you look in industry, it's actually a well-known method if you want to generate comparably low flows and comparably high pressure heads. Because if you connect just two pumps in series, you just add the pressure heads. So this is usually used when you have confined space and need to generate high pressure heads. So we use it a bit differently because we keep more or less the size the same, but we reduce the circumferential velocity, right? By adding an additional stage, looking at the Euler equation here, we can reduce the circumferential velocity by approximately 30% compared to single stage pumps. As circumferential velocity is directly related to the shear stress within the pump, we can reduce the shear stress theoretically by 30% by just adding a second stage to the pump. However, you can also see from this image already that such a multi-stage pump is definitely more complex than a single-stage pump. And this is one of the major challenges we have, of course, with developing an implantable system, the complexity. So that's why we came up with a system which seems feasible to us. So it's a so-called back-to-back impeller configuration. So can you see my the laser pointer here, or probably not, right? Let me.
No, we can't.
So it's on. No, okay, no, that's not the laser pointer. Sorry for that. Now you can see that, right? Yeah, that's the laser pointer. So you can see that we have like two stages, so two sets of blades on the top and the bottom of a rotating cylinder. So we have just one moving part with two sets of blades. So we have an inlet, the first set of blades pumping towards the second stage, and the second stage pumps toward the patient. The size of the impeller is just 11 millimeters, so the entire pump is like 2.5 times 2.5 centimeters, which is much smaller than the clinically available pumps. So how did we make it that small? We opted for a fully levitating impeller, so no mechanical bearings. In radial direction, we have a hydrodynamic bearing, so it's a rotating cylinder within another cylinder, which is not rotating. And axially, we are centered through the reluctance forces of the motor. This is also one of the benefits of such a multi-stage pump, that the axial forces, so the axial thrust, is actually lower than in single-stage pumps because it's compensating. So that's why we can use the reluctance forces of the motor itself to balance the impeller within the pump. So this entire strategy allowed us to miniaturize the pump and reduce the circumferential velocity while achieving the required pressure head. And I mentioned already the pump is very small. The priming volume is like 3.7 milliliters. So to show that the pump fulfills the requirements, we did CFD to evaluate the pump performance. So we used the standard CFD setup with urons and K-omega SSD model. We also used the non-Newtonian Karo-Yasuda fluid method to appreciate the non-Newtonian properties of blood. In the end, we came up after a mesh sensitivity study. We had like 9.3 times 10 to the power of 6 cells. We investigated washout to estimate thrombogenicity through a passive scalar transport model. And we also calculated the normalized index of hemolysis through the well-known power law with the parameters of Heuser-Opitz. We compared the results to the results of the HeartMath-3, not only in the pediatric operating condition, because this would be a bit unfair, of course, because we designed a pump for pediatric patients. The HeartMath-3 is not designed for pediatric patients, but also in the main operating point of the HeartMath-3. To validate these findings, we developed a very flexible model, actually. So it's like a test bench where we can print the pump housing and the pump impeller. We can put it on the shaft. So it's not yet a complete pump with all bearings and motor ends on, but we can just put the impeller on the shaft, rotate it within inlays, forming the fluid volume of the pump, and then thereby assessing the HQ curve. So we can rapidly iterate different versions of the pump. So here are the results. So you can see here the flow path through the pump. So entering here, going through the first stage, then going to the second stage via the crossover, and then ejected or propelled towards the patient. We achieved the design flow rate of 1.5 liters at a circumferential velocity of 3.5 meters per second. So that's already one of the main findings, right? Because usually that's a bit faster, especially in this small pumps. The pressure head was around 55 millimeters of mercury at a rotational speed of 6,400. The efficiency is like 33%. And here in the black signs there the experimental results. So the colored one are the CFD, and the black ones are the experimental ones, and you see a good match between those. So comparing it to the HeartMath-3, we can observe that we have a lower circumferential velocity compared to the HeartMath-3 at at the pediatric and the adult operating condition. We have a similar hemolysis index, so it's not lower. This can be explained by the fact that we use a hydrodynamic bearing to make the pump much smaller, right? Because the HeartMath-3 is quite a big and bulky pump. So it can't be implanted in pediatric patients. So we could interpret these findings that we can achieve the same hemolysis index as the HeartMath-3 with a strongly miniaturized pump. And you see the degree of miniaturization in the washout, right? Because the washout here in green for the two-stage pump and in orange for the HeartMath-3, it's much faster for the two-stage pump because it's much smaller in the end. So in the motor side, we are cooperating with the ETH Zurich. They develop the motor fuzz. So the motor is able to achieve the forces in radial direction and axial direction, and of course the torque to rotate the motor. The estimated losses of the motor were estimated with 0.18 watts. And we then simulated actually the worst case scenario where we applied these 0.18 watts directly on the fluid volume without any wall of the housing or without any surrounding tissue. And we saw that the maximum temperature increases as 0.4 Kelvin. So this is lower between the 2 degrees threshold, which we need to achieve for regulatory purposes. Also from the virtual fitting point of view, we virtually implanted this pump into 7 pediatric patients. So all these patients are either end-stage congenital heart failure patients or patients who received an LVAD and we found that in all these patients, the pump achieves an excellent virtual fit. Although word of caution here, the LVADs and virtual fitting, it's it definitely does not reflect reality. So to conclude, and the next step, so what we showed so far is that this two-stage pump is feasible in terms of hydraulics, bearing, and motor design. We also have now a portable driving unit, which makes animal experiments or blood experiments much easier. Of course, this two-stage pump is more complex than a single-stage pump. That's why we teamed up with a company, Litos. The company Litos, they're experts in 3D ceramic printing. So these components come actually with a great surface and very good accuracy. So at the moment we are investigating the use of this material in blood experiments with this pump. So the next steps are now to assemble these components and go to blood tests. And in the end, our aim is to have some animal experiments, hopefully by next year. Yeah, so I want to appreciate the team behind all this work and thank you for your attention.
Thank you, Marcus. This is a great talk and a very exciting project. I guess a lot of clinicians and patients are really waiting for your results and to come to the clinic.
Thank you.
We move to the next speaker, who is going to be Anna Martins Consta. She is currently a postdoctoral researcher at the University of Twente in the Netherlands, and she is a chemical engineering, membrane science, and medical engineering. Her research focus is on artificial organs, especially in artificial lungs, artificial kidneys, and artificial blood. And she worked on developing on the Renox, a novel combined artificial lung and kidney device for ECMO patients. So we are all curious about your research, Anna.
Thank you very much. Good morning, everyone. Thank you for the introduction. And also thank you for the opportunity to present in this very inspiring tribute to Dr. Bartlett's work. In this presentation, I will go through the development of the Renox device, that is a new artificial lung device that integrates the function of kidney support as well, and developed here by us in the University of Twente with our project partners in the RWTH Aachen and the Medical Hospital in Hanover. And since this is a tribute to Dr. Bartlett, I would like to start by thanking him for his support to new ECMO technology, including the Renox. So in the first year of my PhD, I got the very valuable opportunity to meet Dr. Bartlett in person and stay for some time in his lab. And this provided me with some great exchanges, insights. So I can only thank him and his team for warmly welcoming me during that time and supporting this project and also many others in the ECMO field. So our motivation to develop the Renox project, indeed comes from extracorporeal membrane oxygenation patients, as we see that after bleeding and thrombosis, severe kidney disease is one of the most common complications during ECMO. Up to 70% of the patients that need ECMO also suffer from acute kidney injury, and most of these patients will then require treatment with an additional extracorporeal machine that is able to provide continuous renal replacement therapy to support the patient's kidneys. Today, all existing approaches to treat ECMO patients that suffer from acute kidney injury involves two separate extracorporeal circuits. So this is how it looks in real life. The system requires two extracorporeal artificial organ devices, also separate pumps, tubing that transport the blood across the circuits, and requiring also additional staff members in the hospitals to take care of these separate machines. And all of this brings increased risk for these patients, such as increased risk of bleeding due to the multiple cannulas, increased risk of blood cell damage, also exposing blood to large artificial surfaces, increasing the risk of blood activation and blood coagulation, and as well as increasing hospital costs. So in this work, we aim to provide a new treatment alternative to improve lung and kidney support for ECMO patients in terms of a new device that is able to combine at the same time the functions of an artificial lung, a membrane oxygenator, and a dialyzer, an artificial kidney in a single device. So only one circuit and only one device could be used, hopefully reducing complications related to current treatment. So the overarching objective of this work was to develop the Renox device, this new device, that in one hand is able to provide sufficient gas transfer to support the patient's lungs in the metabolic requirements of the patient's lungs, as well as to provide toxin clearance and filtration in levels that will be able to support the patient's kidneys in comparable levels as what is currently given to them in continuous renal replacement therapy. The development of this device followed different steps, three main steps. So in the first step, we analyzed the clinical requirements, so what was required for this device to support the patients from the clinical side. And then in the second part, we had to develop fundamental knowledge on trying to understand how to combine the functions of gas exchange membranes and dialysis membranes in a single device, in a single membrane bundle. In a third step, then translating these clinical needs and the fundamental findings into the design of our Renox prototype so that in a final step, this device could be manufactured and tested to verify if functions were met. So starting with the clinical needs, we received from the medical professionals that they would like the Renox to be a hollow fiber membrane-based device. So this means that for lung support function, we should use gas exchange fiber mats to supply sufficient oxygen and CO2 in levels able to meet the metabolic requirements for lung support of our ECMO patients, while for kidney support function, we should use dialysis fibers to again support the kidneys of the patients providing continuous renal replacement therapy. Regarding the design of the device, the Renox should minimize as much as possible the membrane area inside of the device as well as the blood priming volume to minimize risks and maximize the benefits of this integrated compact approach of the Renox. And it would be also designed to reduce risks related to blood coagulation and blood cell damage. And finally, at this research stage, it was designed for the Renox to be a modular device that would allow the membrane bundle of the device to be removed for analysis after tests. So in the second part, then we had these requirements in mind to define specifications of a combined membrane bundle able to integrate both lung and kidney support. So our first study then related to the lung support function, and we wondered how we could combine gas exchange fibers and dialysis fibers in a single device that would at the same time minimize device size while still being able to provide sufficient lung support capacity. So for that, we compared the performance of devices that had all fibers open, all fibers transporting oxygen, similar to commercial oxygenators in the market, with the performance of devices that had part of their fiber layers closed, and those closed fiber layers were not able to transport oxygen and would then represent the dialysis fibers in the Renox device. And we tested then the performance of these control devices with our devices with fiber layers closed. And what we saw in our experiments was that devices that had every fourth gas exchange fiber layer replaced by a closed fiber layer actually kept same gas exchange performance. So the gas exchange performance of the ones with closed fiber layers was still sufficient. And this indicated to us that we could replace every fourth gas exchange fiber layer of our device by a dialysis fiber layer, so 25% of the gas exchange fibers by dialysis fibers in this alternating way, while still being able to provide sufficient lung support. So this first study assisted us regarding the lung support function. However, we still had another challenge regarding to the kidney support function. Since in typical dialyzers, blood flows inside of the dialysis fibers, while in the Renox, we would like to have blood flowing outside of both gas exchange and dialysis fibers. So we wondered if you could use commercial dialysis fibers with blood flowing outside of them, while still being able to meet the kidney support requirements. So we then tested these dialysis fibers in these two different ways, with blood flow outside and inside, and we saw that at least for the removal of small blood toxins such as urea and creatinine, that having flow inside or outside of the fibers led to a similar performance. However, filtration capacity in the outside-in mode was reduced, most probably because these commercial fibers, they don't have an outer selective layer, so they are made for the inside-in use. And this indicated to us that we will need dedicated dialysis fibers for the Renox application to improve filtration efficiency, but not only for that, also because fiber mats of commercial dialysis fibers are not yet commercially available or not done in laboratories. So together with project partners, we are developing such dedicated fiber mats for the Renox application, so spools of dialysis fibers can be produced and knitted as mats to integrate our device for future tests. So in the last part of this project, we then translated this fundamental knowledge and clinical needs to the design of the first Renox prototypes. And the Renox device at this stage is composed of three main parts: an integrated membrane bundle, the housing of the device, and the section through which blood flows in the device. In the Vinox integrated bundle, every fourth gas exchange fiber layer is then replaced by a dialysis fiber layer, and this allows us to save total device area, with the Vinox in adult size having a similar area as a single membrane oxygenator, however, aiming for both support of both lungs and kidneys instead of only the lungs. The device housing then seals the different compartments of this integrated membrane bundle to separate the inlet and outlet of blood, gas, dialysis fluid, and filtrate flowing through the device. This design also allows us to have this modular required approach that would allow the membrane bundle of the device to be removed for analysis after experiments. And our partners in Aachen also help us to improve the design of the blood flow path of the device, allowing to reduce risks related to blood coagulation and blood cell damage in the Vinox. So finally, in the last step, we were able to manufacture the Vinox device, now aiming for the support of small adult patients of up to 40 kilograms and working at a maximum recommended blood flow of 3.5 liters per minute. And these devices were then evaluated in blood tests, keeping the dialysis compartment not contributing to gas flow. And still, we were able to see that the Vinox was able to provide sufficient oxygen and remove CO2 in levels that would meet the metabolic requirements of our target patients, even above the maximum recommended blood flow of this device. So in summary, looking back at the requirements, we were able to show that the Vinox met the desires of a compact device with a size comparable to a single oxygenator, but aiming for support of both lungs and kidneys. And this device in vitro test also showed to meet the metabolic requirements for lung support function. However, we still have to evaluate combined lung and kidney support with the Vinox when dialysis fiber mats are becoming available in the coming months. So this study shows that we are developing this new alternative for to provide combined lung and kidney support for ECMO patients suffering from acute kidney injury, and those are steps towards this new treatment alternative. I'd like to thank you all very much for your attention and participation in this session in honor of Dr. Bartlett's work. Thank you very much.
Thank you so very much, Anna, for an excellent talk, and it's good to see what the future will hold. We will move swiftly along, and it gives me great pleasure to introduce Libera Frizelio, who is an assistant professor at the University of Twente. Her work focuses on the development of simulators, merging computational physiological modeling with soft robotic replications of the human cardiovascular system. And her goal is to create high-fidelity anatomical and pathophysiological testing environment to support the innovation of an active mechanical circulatory support systems. Libera.
Hello, everyone. Good morning. Thank you for the kind introduction and also for this honor to present this great event in honor of Dr. Bartlett. So I work in the group of cardiovascular and respiratory physiology that is led by Professor Dirk Donker. And our focus is to develop high-fidelity simulators of the cardiovascular and respiratory physiology. And I will show you today an example of what we are conducting currently in the field of VA ECMO. So the type of simulators we develop are the so-called lung parameter models. They offer a full body representation, so the 4 heart chambers, systemic and pulmonary circulation. We also have recently put a lot of emphasis on the ventilation system, so added the model of chest wall and lungs, and also a numerical model of the mechanical ventilation. That is a therapy that sides VA ECMO and BV ECMO very often. We reproduce what the so-called heart-lung interactions. So the alveolar pressure affects the pulmonary capillaries, and the intrathoracic pressure affects the hemodynamics and the 4 heart chambers and in all the vessels in the rib cage. Now the type of output you can obtain from these simulators are shown in this video. So you can obtain pressure waveforms, pulmonary pressure, CVP, PV loops, and they are affected, of course, by the ventilation mechanics that you observe here in this video. Here is a model of the mechanical ventilation where you can set all the parameters as in the normal ventilator. And we can also play with, we can also observe oxygen and carbon dioxide concentration all over the body. We can play with patient parameters, impairing the oxygenation function in the alveoli, or also impairing the left ventricular function, so creating compromised both ventricular functions and respiration if needed. And then, of course, we can observe the resulting patient status and also then simulate the ECMO therapy with a dedicated user interface. After configuring the therapy in the most appropriate way for the clinical case considered, we can turn on the ECMO, and the simulator will predict the effect that the ECMO will exert on both hemodynamic parameters, but also on respiratory parameters, so both on pressures, but also on oxygen and carbon dioxide. I'll go quick on that for lack of time. So for some scenarios, it is important to test the real medical device. So the computational model of the ECMO might not be sufficient, especially when there are new devices under development and we want to assess their efficacy and effectiveness, also in collaboration with medical industry. In that case, it is important to offer a realistic test bench where we can really observe the performance of the real medical device. In these situations, we use the so-called hybrid in silico in vitro simulator, where on the left side you have the computational model I mentioned you before that represents basically the patient digital twin, as it is called nowadays. But we can also reallocate one or more anatomical components on the hydraulic domain. So in this case, for example, we assume that the right atrium and the aorta are on the in vitro domain, so we can attach a real ECMO and test its performance. Of course, the right atrium and the aorta will communicate in real time with the reminder patient model on the computer. So here is an example of the ECMO attached to this hydraulic machine. In the computer here on the right side, you see the patient model running in real time, so cardiovascular and respiratory functions. And what we can do, I'll go a little bit quicker on the video. Here, of course, we are impairing the respiratory function just to justify that we need ECMO support. And now what the operator can do is just to unclamp the device and start the pump as it will be operated in the clinics and regulate the speed of the device in order to obtain the desired flow. So here you see the flow provided by the device that is increasing as the pump speed increases. And then we can observe in real time the effect that this ECMO flow has on both hemodynamic and respiratory parameters. Worth to mention, we use water and glycerol, not blood, so the effects of oxygen and carbon dioxide is simulated, assuming that the oxygenator works perfectly and calculating then with a mass balance equation, oxygen and carbon dioxide everywhere in the body. Now, mentioning that we have a cubic box for the right atrium and the aorta is maybe not that anatomically correct. So we have now come up with a new simulator that you see in this figure. On the left side is always the patient digital twin, so the cardiorespiratory model with the mechanical ventilation. But on the right side, we have created the patient-specific model of the aorta. And we always have then this communication between the two systems. So one chamber represents the right atrium, so we can cannulate the venous side of the ECMO in a physiologically consistent manner. Two chambers are then used to simulate the inflow and outflow of the aorta according to the residual contractility of the patient and the systemic resistance. And then we have an additional big box in which the aorta is embedded. And this big box represents the intrathoracic pressure, so always to simulate this heart-lung interaction also on the in vitro domain. We wanted to personalize and show that the model can be personalizable. So we used a clinical case scenario from the UMC Utrecht, which is a patient that underwent selectively via ECMO to prevent primary graft dysfunction after lung transplantation. So we also had the CT scan of the patient before via ECMO. We retrieved the aorta geometry from the CT scan and produced a patient-specific aorta phantom. The phantom was embedded in our hydraulic system, and the hydraulic system was personalized with patient's data. So with the ventilation data, the ventilator, so PEEP, peak pressure, respiratory rate, and so on. Hemodynamic data coming from the cardio monitor, so heart rate, ultrasound about the ventricular residual contractility, systemic and pulmonary circulation status. And then, of course, we also tuned the ECMO therapy according to the flow that was reported in the clinical records. So we tried to create the same clinical scenario as in the patient. And also for the phantom, worth to mention, we did not only reproduce the anatomy of the patient, but also the mechanical properties. So we came up with a recipe on how to create phantoms that can replicate a desired target compliance. Here are the results, a comparison between the simulations and patient's data in terms of blood pressure, PAP, wedge pressure, the left ventricular stroke volume for different ECMO flows, and the pressure drop across the oxygenators and the venous pressure of the ECMO. So we really tried to understand whether the simulator could really replicate all these clinical measurements. And also about the ventilator, we also understood whether the simulator could represent heart-lung interaction. So you have here 2 ventilation cycles with insufflation and exsufflation, and you can observe how they modulate the pressure in the aorta, the wedge, the CVP, and the pulmonary pressure. And also this, of course, will finally impact left ventricular and right ventricular stroke work with a very precise dynamic. And this dynamic we observed that was the same as reported in clinical literature. And finally, also the pulse pressure that we observed in the aorta was very similar in terms of trend to what was reported in the clinical literature. So let's say that this could open a window on how to study how to better modulate mechanical ventilation in patients supported with an ECMO. But also the phantom is ultrasound comparable, so we can also study watershed phenomena. Here is an example of an echo PIV conducted on the phantom. On the left side, we have the left ventricular flow, on the right side, the ECMO flow. And here is a video. So during systole, the left ventricle overcomes and the watershed moves downstream. But then during diastole, the ECMO flow becomes more predominant and the watershed moves upstream. So the idea is to use this simulator to support medical technological innovations, of course, but also to create new therapeutic strategies, clinical decision support system that can better tune ECMO therapies and mechanical ventilation according to patient scenarios. We also want to go beyond model personalization. We want to create also virtual patients ad hoc to challenge new technologies in particular case scenarios for which safety and effectiveness might not be that obvious, and hopefully to produce evidences that can complement evidences coming from traditional clinical trials. Our simulation platform can be also used for other type of medical devices, such as LVADs. And for this, my conclusive slide is just to mention the study that we are now conducting with the FDA. Is an interlaboratory study to which 16 laboratories worldwide participated. All laboratories with an in vitro setup to test an LVAD. They all tested a HeartMate 3 device, and we collected all the results. And the idea is to come up with a consensus and share methodology on how to assess reliably an LVAD in vitro, so that in the future we can use this test bench as validated tools and recognized tools also by regulatory bodies. And so produce evidences that can be effectively used to approve these devices, at least from the hemodynamic point of view. And this concludes my presentation.
Thank you so much for an excellent presentation. So we've got about 10, 12 minutes for any questions. So any questions, do feel free to pop them into the chat or into the Q&A, and we will try to address them. I might kick off for Thomas. A really practical really practical problem that we all face. And it's great to see your work on the axial stresses loaded. As a clinician, I guess one of the things that I really worry about is particularly the rotational and Jutta will laugh at me, the torsional forces. I'm not sure which what you call them, particularly for the neck as the patients sort of move their head around. How would you go about exploring that in the same way that you have for the axial forces?
Yeah, so great question. As I said, I mean, this was the first, study so far. Nobody tried to assess this, to be honest. I think we're working currently also to have more, models which could simulate. This is a little bit tough, of course, to have really, because we started, like with suturing pads, etc., which the surface is not similar to skin. That's, clear, and therefore we came up with these slaughterhouse porcine models. We're investigating this if we can also simulate really like the neck. It's really hard, to be honest. I think the combination maybe with force measurements in the clinical arena is also what we're thinking of, to do it really in a clinical setting. I think that's what we have to do to really clearly make a recommendation, let's say, what is best. But for us now, I know, and I think most of the centers are doing, we are combining in the clinical arena now really like an adhesive with sutures, and I think then we have both the benefits of the stiffness of both, let's say, materials. That's what we should do.
Jutta.
So I would also have a question on that, Thomas. Are you somehow, do you have an idea how you could somehow also see the effect on the tip position of the cannula? Because later on we'll hear a talk of Lisa Prahl, and she shows in a simulation that if the cannula moves within the blood vessel, that tremendously changes the flow within the cannula. Do you have an idea how you could test that in this kind of porcine model?
Yeah, really tough, I would say, as we're not really now simulating the blood vessels. Then I think we would have to do this maybe really in a in vivo model in animals. So that's really tough to simulate. So far, it was really, but I'm open to any suggestions. Let's say first idea how to measure this, because as I said, so far we had no clue how much forces are really applied to the cannulas. For us, it was really, as I said, very impressive that there are so big variations with sutures even within one surgeon. To be honest, this was surprisingly for us, and that sometimes really the cannulas were let's say almost damaged because there is too much stress on the cannulas, and this could, as I said, limit dramatically flow and lead to low flow conditions. And every movement, what you just said, that's why we don't want to have any movements. If the position inside the vessels is not optimal, then there are more shear rates and really poor washout. And I think that's the main problem. We only see the tip of the iceberg, so to say. Then we have maybe infections or dislodgements, but inside, I fully agree, we don't have a great solution so far how we can simulate this or measure in a better way.
Perhaps there's an option to team up with Libera if she could perhaps add that to her mock loop, her physical mock loop, that you really could place cannula in the vascular system.
Yeah, that would be good. Of course, our next step is to have branches of the aorta as well. So what you saw now was a very simplified aorta. But of course, that's what we are currently doing as next step. So perhaps we can help you.
Yeah, I was thinking about suggesting exactly the same, that you should team up, you guys, and come out with a solution for us clinicians. And again, for Thomas, I think building up on that topic, I think the micro movements, like the rotation or the micro movements on the skin entrance of the cannula, that's a big issue for infections as well, but also for bleeding. Like that we cannulate and we see the patient is not bleeding, and then of course coagulation is changing over time, but also this micro movement. And as a surgeon, I'm called to put a stitch on the entrance point of the cannula because the cannula is moving. So but the stitch is also creating ischemia on the skin if you don't put it right. So I think these are all topics that you might want to address with your next experiment, or when you move to the clinical side, or maybe you already had some evaluation and maybe you come up with some suggestion for surgeons how to deal with the entrance point of the cannula.
Yeah, thanks, Silvia. I think still for the entrance of the cannula, I think it's still the stitch. Just the adhesive is not enough. So I would go for, that's what we also do now in our clinical practice, one stitch at the entrance, and then we do 2 fixations with securement devices. That's what we currently do.
And I think also it depends on the way you cannulate the patient, if you cannulate percutaneously or if you cannulate surgically, semi-Seldinger technique. And sometimes the surgeon puts stitches inside the wound and you don't see it. So that can also cause, when we decannulate, we see the damage on the vessel itself. So those are all questions that we have and we still need answers. And maybe you will help us finding answers. Also how to cannulate surgically and do a perfect hemostasis on the vessel itself. And I don't know, Nicholas, if you have experience or suggestions for your surgeons.
Yes, I find suggesting suturing to surgeons works extremely well as an intensivist. Jutta.
I would have a question to Marcus. Marcus, I think it's a fantastic idea to have this dual stage pump, as you rightfully said, that the low flow with high pressures is giving us quite a headache as engineers with pumps. Have you, it looks rather big with this transition bit. Could you comment a little bit on the dimensions of the pump?
Yeah, so in the end, it's just 2.5 times 2.5 centimeters. So it's actually pretty small. So it's way smaller than like the Hadmet 3 or so. So the duct, it looks big on the slides, but it's like 6 millimeters in diameter. The impeller is like 11 millimeters in diameter. So it's pretty tiny.
Yes,
The
that
device. So
sounds tiny indeed. That's always a problem with these 3D models on the screen. They always look
exactly. So the CADs and this 3D models, it always looks big, yeah.
Yes.
Yeah.
Do you, I mean, you're creating different pressures. So is it really enough to have the magnets around it to keep the rotor in place, that it's not moved away from the higher pressures?
Yeah, that's the good thing about the two-stage pump, right? Because the pressures acting on the impeller are just half of the pressures which would act on a single-stage pump, because you develop half of the pressure in the second and half of the pressure in the first stage. So we just have half of the axial thrust. So we are not concerned about the axial thrust. What we are more concerned about is the acceleration of the patient, right? Or the motion of the patient. So this is way higher. So also for the radial forces, right? For all the forces within a blood pump, if it's durable and not like next to the patient. So these are the real challenges, right? So, and at the moment, we're actually measuring accelerations in pediatric patients to see what accelerations are expected. So our kids are jumping on the couches, and, and from the couch and on trampolines to really learn what the accelerations are we have to cope with and which forces we need to develop. But at the moment, it looks reasonable.
Okay, so you're
yeah.
more concerned about the kids playing around and being on a swing or anything like that.
Exactly.
Okay.
So we are way
So no
more
bobby
concerned
cars.
about that than exactly. So we are way more concerned about that, yeah, than about the hydraulic forces. Yeah.
Okay, thank you.
Yeah.
Brilliant. Sylvia?
I also have a question for Marcus. Again, as a clinician, as a surgeon, I'm always thinking about 3D and fitting and implanting things. And I was really excited and surprised about the fact that you tested your pump and you simulated the virtual fitting was done on patients from age 0.9 to 15 years. That's quite a big range. And a 15 years old patient is almost an adult with quite a high required blood flow for for the whole body. You were talking about a flow rate of about 1.5 liters per minute, but would you expect to, create also a pump that will fit for older patients, like 15 years old, to provide higher blood flow?
I mean, it's a very good question. So at the moment, I think with this pump we can probably generate 2.5 liters, maybe not much more. Then we would need to make it bigger. But what we are thinking of is a partial support option, right? Also because in terms of a business case, because a pediatric pump is really hard to find funding for because of the limited patient numbers. So you always have to provide a business case, and I think partial support for bigger patients or adults maybe exactly this business case, right? That we claim we can also support these patients, there's less sick patients with 2 liters per minute, and with a very hemocompatible pump, right? And the same pump could be used for pediatric patients. So that for small pediatric patients, right? Those in full support. So that this would be our business case and our strategy to go into the market.
That's opening a very, very interesting field for adult surgeons as well and adult patients. Nick.
Lovely. So my question's for Anna. I love the idea of combining ECMO and renal replacement therapy because it's such a common combination in our clinical practice, with about a third of patients needing both. My question is, have you started to look at the different dialytic and filtration modes and how they can work, particularly with some of the dialysis flows we get? So we would routinely run a dialysis flow of 2 liters, but we may go up to 3, 4, or 5 liters for some of our sickest patients, particularly those having very high dose ultrafiltration with toxicities and other things. What range of dialytic flows do you think that you can get out of the combination therapies?
Yeah, thank you, Nikos, for the question. So we are very curious as well to start testing this dialysis conditions as well as all the kidney support modes. So we expect with the Venox to be able to provide all of the kidney support modes, so dialysis plus filtration or diafiltration. However, since we don't have the dialysis fiber mats yet, it's hard for us to assess what would be the interaction with the whole system, right? So how pressures would change when we have the integrated membrane bundle. While having such high dialysis flows, for instance, pressures would also change in the dialysis fibers, and because they're permeable, this would also impact our gas exchange. So although we expect that we could be able to provide doses that are similar to what is currently given in continuous renal replacement therapy. So I believe for adults is around 20 milliliters per kilogram patient per minute. So we expect that we would be able to provide that. However, we cannot really assess that yet because using only fiber, as you were saying in the experiment, using single fibers in a dialyzer will be different than using the fiber mats in the Venox, and also the configuration will be different. The flow inside or outside will differ. So I hope next time in next presentation we'll be able to answer that with the mats that are being produced by our partners.
Thank you very, very much. I think there is plenty of other questions to be asked. Unfortunately, I think we've also just run out of time. So I think I might close this particular first session down. Thank you very much to all of our speakers. It's been a fascinating introduction into what is the future of ECMO and what comes next. And it's wonderful to see such a wonderful collaboration between our clinicians and engineers. And with that, I'm going to hand over to our current president of EuroElso, Mirko Belliato, chair of our scientific committee, Justina Swoll, and our treasurer, as it turns out, for EuroElso, Apana Hoskoti, to run the next session. Thank you all.
Thank you, Nick.
Co-hosts.
Thank you, Nick. Thanks for the kind presentation. As I mentioned, I'm Mirko Belliato and come from Italy, Pavia, Foundation San Matteo. And I'm lucky because I am the chair of the European branch of EuroESO. It's time for the next speaker, Lisa Prahl-Wittberg. Lisa Prahl is a friend, first of all, and secondly is a very, very good professor in fluid dynamics at the Department of Engineer Mechanics at the Royal Institute of Technology in Stockholm, Sweden. She has a lot of experience in the ECMO field, and she focused the study on fluid dynamics of cannulas, oxygenators, and pumps. So, Lisa.
Thank you, Mirko, for the kind introduction, and also thank you so much. I'm super happy and thankful for being able to present here today in the memory of Bob Bartlett. So let's see if I can. See if I can get this working properly. So the topic on my talk today is understanding your ECLS devices, current myths and misconceptions. Just to get started, due to time, I will not be able to focus on more than a couple of the devices, but I'll take a look on cannula blood pumps and tubing connecting outlets. As we all know, the thrombogenetic potential of the ECMO circuit is linked to mechanical characteristics, and we know from the study by Sean et al. A couple of years ago that the shear stress and the exposure time is important when it comes to blood damage thresholds. So here we see the shear stress versus exposure time and onset for hemolysis, platelet activation, or von Willebrand degradation. And what we see here is that exposure time and shear stress is equally important. So even if we have a low shear stress, the exposure time to the stress is something we need to pay attention to. So high and low shear stress exposure time, we should try to keep the stress about below 12 pascals. That in relation, we can say that in the body usually would be about 1 pascal. But we also know that von Willebrand factor may, in an elongational flow, when it is more pulled, may unfold already at shear rates about 2000. That would be a pascal of like a stress of 6 pascals instead. So that's actually lowering our threshold slightly. But if we go over to the cannulas, is it just a pipe? Will it drain evenly through tip and side holes? Is it better to go for a smaller cannula diameter than a larger one? Let's start with the first one. It is a pipe, but it's quite a complex one. So it is a highly dynamic flow that we need to pay attention to. And sometimes I think maybe we forget how complex this actually is. Looking at the drainage, this study comparing a multi-stage cannula in MRI and computational fluid dynamics, we see that the drainage is carried out mostly in the most proximal holes and then lowering as we move towards the tip. However, if we move from a generalized geometry to a patient geometry, this drainage fraction is actually shifted. So then instead, we may drain a little bit more through the tip than through our other holes. So there's quite a dynamic situation here where the drainage may alter depending on the geometry that the cannula is positioned in. And what is the reason for this? Well, if we make it basic, we see that in the vessel flow, we have a velocity that is 1/10 of the velocity in a cannula. That means that the pressure gradient over the side holes is larger just at those proximal holes, and therefore also inducing this drainage. So the pressure difference will play a role, and that's going to depend on the vessel geometry, cannula design and positioning, as well as the flow rates in the cannula and vessel. Also, another topic or aspect of this is the fluid available to drain. So for example, if we would drain lower than what we have available, but at the same time we are obstructing the vessel, that means that we have a pressure buildup just around the cannula, and then we may also alter a little bit the draining characteristics. For the cannula diameter, then we're just going to I'm just going to bring it down to some basic numbers. If we decreasing the diameter, we will increase the velocity. And by increasing the velocity, the Reynolds number, which is sort of assessing or giving a sense of the unsteadiness of the flow, will increase. So taking this all together, the increase in velocity will make the pressure losses increase because it's dependent on the velocity squared. But also the shear stresses with an increased velocity will, because of the shear rate, the du, dy, will also increase. So there are several aspects here one needs to take into account in order to say which one is actually better. Moving to the pumps. Oh, yes. So this was more summarizing. But moving to the pumps, is it one pump that fits all of it? We usually see that the pumps are operating in a large range of flows. Is it true? And it's also sometimes said that lower pump speeds are better. So what do we know when it comes to pumps? We know that we have a highly unsteady flow. At the blade tips you see here, the coloring gives you a little bit of a sense of the differences in the flow rates, or the velocities rather. And also the red region says that we have recirculation zones, so that's stagnant regions. Above the impeller, we will, for some pumps, see a coherent structure that is moving above the impeller and actually against the incoming flow up towards the inlet. This flow situation is usually more prominent for certain designs, but as well for lower flow rates or flow pump speeds. And we also, again, have some coherent structures in the impeller base. So this is what is hidden in the flow when you're running your pumps. So generalizing slightly, then high flows will increase unsteadiness, lower flows or RPMs will make the flow slightly more stable. We have more stable structures. So that means we are getting increased shear stress level for the higher flows and increased residence times if we run it lower. And bring back to the figure that I showed in the beginning, then again, just to point out that we need to work with both of this, and it's going to be difficult to get one design that encompasses all of this and lowers both of them, because they're going to work in parallel or against each other. If we look on the literature, just comparing, this is computational studies on adult and baby pump, we see the activation of the platelets are about the same independent if it's an adult pump or low flow rates. Residence time is increasing for lower flow rates. And this is also something that was shown in another study where the middle figure shows that the recirculation time, that is rather the time that the blood resides in the pump, increases at lower pump flows. And also the hemolysis is increasing. And here we can also see on your left-hand side that the hemolysis index predicted by computational studies and experimental studies are quite different, but the trend is the same, and that's an important message. So really, what we need is pump design specifically for the flow rate regimes that they are intended to operate in. Come back to the session earlier this morning, and it also calls for revisiting pump design for particularly low pump speeds. And then it's also what we don't see in these ECMO circuits. We have a lot of different components that has small steps in it or protruding objects into the bloodstream. And with the fact that blood is opaque, we don't really realize what that does with the flow. So this is a study, the one with the connectors from the 2017, the group of Ku et al. And there you see the steps where you have a strong recirculation bubble formed, which is also prone for stenosis or thrombus formation. And the right-hand side figure is a membrane lung outlet, where you see it's pretty clear how these objects that are protruding into the blood flow is causing recirculation zones, but also you see the Luer lock where you have a stable recirculation up there. So at the right flow conditions, whatever right is here, any obstacle would cause flow separation, and that is a potential risk of coagulation onset. So it's more of we should think of how we can minimize these type of design features in our devices. So with that, I'll come to a summary or end of my talk. We have devices that are designed for a smaller range of operating points. As soon as we move outside of it, we'll find some maybe unwanted features. They're highly dynamic. We have a balancing act between high shear, low exposure time and low shear and high exposure time that needs to be taken into account at all times. There's some differences in our in vitro and silico in vivo that we need to be aware of when we're considering these studies that are out there. I just showed a snapshot of it. And I also want to say that we need to be a little bit aware of this studies out there, because average values are not always giving the full picture, because this is such a high dynamic, like a dynamic flow situation that we're dealing with. And with that, like meetings like this, when we are meeting across disciplines, are super important, because we need improved understanding from all angles for improved device design. Thank you so much.
Thank you, Lisa. Aparna, please.
Hello, thank you everybody for joining in this 24-hour virtual summit in the legacy of Bartlett. I now have the pleasure of inviting Mike to give us his talk. Mike Broman is an intensivist and ECMO specialist working in Karolinska, a world guru in transport of children and adults on ECMO. And I'm told he loves to slurpee oysters. So Mike, the floor is yours. Tell us about the transport of ECMO in children. Thank you very much.
Thank you for this honorable invitation. Can you see my screen now?
Yes, we can see your screen, yeah.
And now it's coming full, I hope.
Yeah.
Yeah. Okay. Yeah, thanks again, and good morning, everybody. It's really an honor to be here. I'm going to talk about transporting the child on ECMO, more like a practical guide in like 9.5 minutes. These are my disclosures. Yeah, we know since a decade that for sure the higher the number, annual hospital treatment number of ECMO, the lower the mortality in all age groups. And this was published just before the COVID-19, where kids reported centers, adult or pediatric, performing less than 30 annual cases. They have an odds ratio for death 3 to 4 times higher than that of a dedicated pediatric center that performs more than 30. So that's a strong indicator. You could do this hub and spoke or regionalization of ECMO treatments on a national regional level or international regional level for improved survival and more efficient resource utilization. If we go for the practical guides, I would say train your teams for safety, mostly simulations outdoors with ambulance and aircraft services, because you will work quite tight with them. You can do water labs, wet labs, water drills any day of the week. At home, you create a noisy, narrow space environment, and it could be really realistic for sure. The equipment you transport on will at least in part have to be customized for transport, but it's very important to know that equipment and know what to find in what bag, etc. Use checklists not to forget important stuff that you need to bring with you. Communication is very important, and try to do this in close loop and really practice on that when you have your simulations and wet labs. Timeouts before every transport item is very important, so everybody in the team knows what's going on and puts emphasis on red flags, so everybody also in the team knows what might be a crucial item. Use an ECMO ABC that's very close to a patient ABC, but it gives you total control over awareness of the whole team, what's going on with ECMO circuit, also for fault diagnosis. and always have a backup plan if you don't reach your primary goal. This is just one example of an ECMO ABC, and you can create your own, of course. And it's the first item is to have power on your console that creates, you have an RPM, is that correct? The RPM, does that create pressure gradients over the circuit? Not all consoles will give you the P1, P2, P3s, but at least does the RPM create a flow? And is there flow within a set limits? Is sweep gas flow on? Do you have a sweep gas pressure? Do you have gas, oxygen from the wall or from a bottle? Is power on the console, the blood warmer? Was your power source wall, UPS, internal batteries? Is the blood warmer on? Do you have the correct set temperature? Is it connected to the membrane lung? Check color of the tubing and temperature. Do you have chattering of the tubing? That will sort of indicate the drainage problems, and at the end, the proper alarm settings. This is very good to use as a mantra. We do that, and it's performed after every disconnection from or connection to the wall, movement of a patient from bed to CT table, etc. This is a standard procedure that you may apply after any intervention or emergency, and it's actually an excellent tool for fault diagnosis. You save a lot of time doing it this way. This is adapted equipment, a titanium stretcher based on the life port, but even though it has everything on it, if you transport a neonate or a kid, just the electrical plug to the wall weighs less than the original life port. Here are others transport carts we use, and they fit in any vehicle. You win a lot if you pre-pack the bags. You have all the stuff with you, like surgical equipment, we manage ourselves. That's a good intention to have out there. Of course, you need checklists for this. Is it the correct packing, etc. But it's also important to know that you carry around a lot of weight. It could be like 250 kilograms, 500 plus pounds, and it might limit your abilities to use, for example, small helicopters to get out to the patient. But even harder when you have to get the patient weight with you back to your hospital with all your equipment too. If you go international or out of state, see to that you bring compatible gas connectors and electrical plugs, otherwise you might end up in a problem. And for longer trips, oxygen supply, calculate that and bring extra oxygen, maybe twice of what you believe you will need. And for power, we always use an uninterruptible power supply, but it's good to know and you have to realize that it's probably not allowed to connect it to the aircraft's electrical circuit. So as soon as you have a chance, try to use resources wherever you are to save your own. That will be electricity and oxygen from the transport vehicle or in the hospital you are, because half an hour later you may be stuck in an elevator and then you have to use your stuff. This is something I learned all the way on the transport. The lid of the flow probe popped up and then we lost flow by 20-25%. Found just closed it and everything was set, but 2 minutes later we had the same problem, but then the flow decreased faster and even more. It was not the lid, it was the oxygenator that started just to clot, so we ended up in an emergency. And since then I always wrap a tape around the flow probe, so I know that that's probably not the cause for flow disturbances. When you're en route, one risk situation is unloading, loading between different transport vehicles. The team leader has to take charge and lead, and it's in our system, at least the ECMO physician. It's important to have a timeout before every maneuver in this, as in any other case. But the thorough walkthrough of each and every step is crucial, because you may have help of staff at the airport, for example, that don't know you, don't know ECMO, don't know anything. And then it's wise to use like a very efficient tool to convey information like the S-bar, and also tell the backup plan. If we don't get into the aircraft, then we have to go back into the ambulance, reconnect to electricity and oxygen to get to stabilize the patient. And in this, the ECMO ABC is very good to use. Climate issues, of course, up here in north of Europe, fog and sub-zero temperatures, you risk to have stopcocks and IV lines freeze in 10 to 30 seconds. Newborns reach moderate hypothermia within a few minutes, even though the heater's on full. So try to protect the kid from convective heat losses. Timeout, ECMO ABC is crucial in this. The baby to pod 2 is a very effective way to protect the kid from heat loss. The baby or smaller child is strapped to the pod with a harness, and you can have heating pads inside. It eases transportation quite a lot actually and protects the kid. But of course, you see the tubing here. You can put a cloth wrap or protect that against cooling too. On secondary transport, and Kenyon may have been there for weeks. And when I go for secondary transports, I always have syringe, saline, and stuff in my pocket if I end up in a situation where I might need to use Fogarty catheter to sort of cleanse cannula. It has not happened on transport. However, oxygenator clotting I've experienced. If you go out of states or secondary, in most cases international transports, check before entering another country's state which medications you can bring. In Thailand, you will end up in prison if you bring morphine. Check liability insurances on site and if you're even allowed to perform medical work on site. Different transport vehicles in our system. This is not the US where you can use a C-17 and play soccer on the way out to the patient. It's much more narrow. You have to adapt. And also in this, when you encounter transport vehicles or ground ambulances you've never seen, check what resources they have for electricity and oxygen. Also, how you, in the most safe way, can load the patient into that vehicle. And that also means that you have to secure all your IV lines, three-way stopcocks, etc., that hangs around to make sort of your equipment or the ECMO circuit quite sort of condensed so you don't hook up in stuff. In helicopter, it's extremely vibration, lots of vibrations, lots of noise. It's very hard to hear alarms. It's hard to communicate even if you have headsets, etc. So now you have to play with your eyes all the time to keep control of what's happening or not happening. So to conclude, well-trained staff and properly adapted equipment, checklist, timeouts, backup plan, use the ECMO ABC, do that inward, do that with your team all the time. It will give you a lot of benefit. Check the transport vehicle before loading, electricity, oxygen. Communication is crucial. Avoid hypothermia, and it's extremely important in the neonates and smaller kids. And if you go international, don't forget the adapters, bring the oxygen, check medical legal issues before you go out there, and of course, vaccination card, passports, etc. Thank you.
Thank you. Thank you, Mike, for this excellent presentation. And I have the pleasure. Are there any questions? I cannot see anything on chat. Maybe at the end of the session. So my pleasure is to introduce the next speaker. It will be Peter Schlanstein. He was affiliated to the Department of Cardiovascular Engineering in Aachen in Germany for the last about 10 years, and his research focus was oxygenators, ECMO, and artificial lung. Now he is a co-founder and CTO of HBox Therapies, where he is developing the next generation of artificial lung technologies. And it's our pleasure to have you, Peter, on this panel. Please introduce us the new technologies and your ideas for the future. Thank you.
Thank you for the kind introduction. Can you see my screen?
Yes.
Thank you. Yeah, I'm very honored for being here, invited to give this little talk with my view on hyperbaric pressure conditions in ECMO. In honor of Professor Bartlett, whom I had the privilege to meet personally many years ago and who supported us also in the early phase of our startup. HBox Therapies is a startup from the University Hospital in Aachen. We started a couple of years ago and we are still headquartered in Aachen. We are surrounded by many great companies and innovators in the ECMO field. And the topic of the presentation fits very good to our startup because at HBox we are developing products on two sides. On the one hand, a next generation membrane lung, which actually has a size that is also interesting for pediatric ECMO applications we heard about before. And on the other hand, we are working on novel gas controller for ECMO-like applications, and hyperbaric technology also plays a role in it. To introduce the topic, I wanted to start with a very normal ECMO setup. We have the drainage cannula, the pump, the oxygenator, and the return cannula, and a sweep gas blender. And we need some kind of pressure to drive the blood flow and some kind of pressure to drive the sweep gas flow. And if we now look at, for example, VV ECMO circuit here, we can look at pressures in the different sections of the circuit. The red line shows exemplary blood pressures present in the circuit. But since we are now talking about gas pressures and partial pressures, we have to add the atmospheric pressure. So I will lift this curve a little to this atmospheric pressure. And if we now starting flowing pure oxygen into the oxygenator, the blood is approaching this value of partial pressure, so roughly 70, 150 millimeter mercury PO2 on the gas side. And if we're now looking at partial pressures in the blood, we come from the venous side, for example, with 50 millimeter mercury, and in the oxygenator, it raises to fully saturated state and, for example, 200 millimeters of mercury. The PO2 values here are only examples. The real life numbers always depend on inlet conditions, blood flow, oxygenator, and other factors. So this is normal ECMO mode. If we now look at the membrane lung in closer, we see a normal situation there with the gas flow and blood flow and the gas exchange. What is important is that the blood pressure is always higher than the gas pressure, at least in microporous membranes. This is something that's should always be ensured also in ECMO. Otherwise, there's a risk of bubble formation in blood and clotting and so on. And from the oxygenator side, this is usually ensured by limiting gas flow and thus gas pressures. But if we make sure that this condition is always fulfilled, then we can also increase gas pressure in a controlled way and use it. And if we now look on the right-hand side, we can see a schematic of a controlled increase of gas pressure. And with that, we can increase the gas density inside the fibers. And with that, also the diffusion driving pressure for oxygen. And this means, for example, we could transfer more oxygen into the blood through the same membrane surface area. Of course, only in physical limits of what can be dissolved and bound in blood. Or we could also achieve the same oxygen transfer with a smaller membrane area. So this is a basic idea behind hyperbaric conditions in a membrane lung. And if we start now from this normal baric conditions, we can look into, I would call it the first level of hyperbaric operation. We can increase the gas pressure in the membrane lung by using some kind of resistance or valve at the gas outlet. And we can increase the gas pressure, still keeping the gas pressure below the blood pressure. And with that, we can increase oxygen transfer and partial pressures post-oxygenator. And if we now, for example, increase the blood flow in the circuit, or from the beginning, we use the smaller return cannula, this would increase the blood pressure even more. So we could also increase gas pressure further. So calling this the first level of hyperbaric oxygenation. And if we go one step further, I would call that second level. And for that, we would actively influence the blood pressure in the oxygenator. That can be done with some kind of additional resistance or backwards pump or another hemocompatible solution. And if we do that, we can control and increase the blood pressure locally in the oxygenator, and this allows us to further increase the gas pressure while still keeping gas pressure below blood pressure. And if we do that, we can control the process in the device and can reach higher gas transfer rates and post-oxygenator partial pressures. So overall, this shows the theoretical concept behind hyperbaric blood oxygenation. And of course, it has to be translated into technical solutions and controlled safely to make it usable in ECMO. This is an example from another group in Aachen around Professor Mutagi and Spillner and Dr. Kashefi. The question here was whether purely diffusive membrane materials like solid silicon membranes or silicon-coated membranes can benefit from hyperbaric oxygenation. And they tested this in bench gas transfer experiments comparing those fiber materials. And they found that already at 40% increase above atmospheric pressure on the gas side, this transfers in nearly 30% more oxygen transfer. Here in this graph, it can be seen. And all, like that, smaller membrane area can reach transfer rates like larger membranes areas. This is seen here in the graph below. So this is a nice example from another group with, they used hyperbaric condition to enhance the performance of new membrane materials. As far as we know, there's no clinical data for hyperbaric oxygenation yet, but this paper from Berlin, I think is interesting because the authors looked at how much oxygen is delivered in a dissolved way during regular ECMO. They looked at ARDS patients on VV ECMO. And what is important, I think, is that the average hemoglobin was relatively low, 8.7. I'm not a clinician, but I think it's typical for those patients. And then the dissolved part of oxygen in the blood gets more and more important. And they were able to show that about 22% of extracorporeal oxygenation was realized as dissolved oxygen. In a patient with severely impaired lung function, this contributes up to 28% of total oxygen uptake. And in their patients, the post-membrane PO2 was around 430 millimeters of mercury. So if we use hyperbaric technology and increase that even further for up to 700 millimeters of mercury, that could allow us to reduce the blood flow or potentially also cannula size or even membrane area. To show the performance potential of this technology, besides other tests, we did a test on a very small commercial oxygenator, the neonatal Highlight 800 LT, which was modified so that we could use it for the hyperbaric technology. And we also modified the blood to hypoxic inlet conditions, so very low oxygen load at the beginning. And we actually ran the device at higher blood flows than it's normally intended for. And as you can see, without the hyperbaric oxygenation, the device is expected, as expected, limited to low oxygen transfer rates. But when we switch these hyperbaric conditions on, the same very small membrane can fully saturate hypoxic blood, even up to 2 liters per minute. To summarize, the hyperbaric approach allows us to, let's say, shift up a gear in oxygenation. You can, for example, expand the rated flow of an already existing device. You can fully oxygenate higher blood flows with that. Or if you start your therapy with a smaller circuit and a smaller membrane, and during therapy you need higher blood flows and oxygen support, you could increase extracorporeal blood flow and extend the support. Or you could think of if you have loss of membrane performance over time due to clotting or clogging or degradation of the membrane, you could maintain the rated blood flow. Or as I showed, use it for new membrane materials. We implemented the hyperbaric oxygenation in a closed loop control system in our prototype using a post-membrane PO2 sensors, blood pressure and gas pressure signals. And if we then switch the device on, the hyperbaric, it can be controlled. We can give a target value in this example on the right-hand side, 750 millimeter mercury. And then it regulates to this target value. And so from our perspective, the technology has shown to be technically feasible, and we are now working on integrating it into our medical device. And yeah, with that, thank you for your attention. And if you're interested in our work, please feel free to reach out after the summit.
Justine, you're mute.
Definitely. Thank you so much. So I have seen some comments in the chat, but this is about the previous presentation. So thank you, Peter. If there will be any questions, we will come back to you. So it's my pleasure to introduce the next speaker, the next presentation. It will be Isabel Esslinger. She is a mechanical engineer and PhD candidate at Charité in Berlin, Germany. And her work focuses on platelets adhesion on surfaces relevant for cardiovascular devices. The title of her presentation is Influence of Material Surfaces and Coatings on Platelet Adhesion in Ventricular Assist Devices. So please, Isabel, we are curious about your presentation and research.
Thank you very much for introducing me. I'm not able to share my screen unless Peter Schlanstein would remove his sharing. Otherwise, I'm blocked. Ah, yes, thank you very much. So now I can. Yeah. Okay. You should see it by now.
Yes.
Okay. Great. Thank you for introducing me again, and also thank you very much for giving me the opportunity to speak at the summit today. I will talk about the influence of different material properties and coating strategies for ventricular assist devices, or especially on rotary blood pumps. So thrombus formation has different causes, like, for example, high shear forces, which leads to wide thrombus formation, also stagnation flow areas with low shear forces, which leads to red thrombus formation. But another important factor to consider is the contact of blood with foreign surfaces. And as we know, in our vessels, blood comes into contact with the endothelium, so endothelial cells. They're antithrombotic, they release antithrombotic substances. So no problem with thrombus formation occurs here in the physiological state. And however, when in contact with an implant, different processes can occur, like, for example, the activation of the intrinsic coagulation cascade, and also proteins like fibrinogen, van Willebrand factor, and albumin adsorb on the surface within seconds to minutes. And these properties are, of course, driven by surface properties. And during this process, a very interesting effect occurs. It's called a Vroman effect. So first, there's a rapid absorption of small abundant proteins like albumin, for example. They diffuse faster and reach the surface first. And secondly, there's a gradual replacement by larger proteins, like you can see here by fibrinogen, for example, or van Willebrand factor. They have a high binding affinity, but they diffuse more slowly to the surface. So they displace the initial layer due to stronger surface interactions. And as you can imagine, the composition of the initial protein layer directly affects the subsequent cell attachment. And especially platelets play a crucial role in thrombus formation. They adhere to the biomaterial through the interaction with the adsorbed proteins. They get activated, change their shape, and also release substances that further activate and recruit platelets to form this thrombus on the surface. And yeah, as I was talking about it before, these mechanisms are influenced by the surface properties. And for surface properties we talk about are, for example, the chemistry, the topography, wettability properties, and surface charge properties. And as you can imagine, these surfaces are not these properties are not independent from another. They also influence one another. So, for example, when we look at wettability, we know that hydrophilic surfaces generally show better outcomes than hydrophobic surfaces. There are also special cases where superhydrophobic surfaces can be good in terms of antithrombotic properties. Yeah, but generally hydrophilic surfaces are associated with better outcomes.
When we look at charge, for example, there is evidence that negative surface charges correlates better with hemocompatibility, also because a lot of blood components are charged negatively, so that has a positive influence on hemocompatibility properties, and discharge can be quantified with the zeta potential, for example. And so what happens is that if a solid surface like a biomaterial comes into contact with a liquid like blood, a surface potential arises, and an electrochemical double layer of immobilized charges or counter ions form at the interface, and the zeta potential marks the boundary between the mobile and immobilized charge, and this zeta potential is measurable with the streaming potential measurement. So it's a measurement method to quantify the surface charge of a solid surface in contact with a liquid. I was talking also about topography, so we need to differentiate between stochastic or, let's say, chaotic roughness, which can be determined by manufacturing and finishing processes. So generally higher roughness values, especially on a micro scale, lead to increased specific surface area, and so there's more surface for proteins and cells to attach, and the protrusions also mechanically trap proteins and cells. And other approaches are also controlled, so micropattern surfaces. So are designed especially to improve antithrombotic properties. For example, by reducing the area that is accessible for platelets, you can influence the wettability properties with different patterns, and also cells can detach through the washout effect, which is also managed by these different patterns. So for ventricular assist devices or rotary blood pumps, usually titanium alloys are widely applied due to different properties like biocompatibility and fatigue resistance. And especially the medical grade alloy, which is a combination of titanium, aluminium, and vanadium, is often chosen for rotary blood pumps due to the increased strength and fatigue endurance. However, these materials have quite low wear resistance, which I will talk about again soon. When we talk about coating strategies, we differentiate between active and passive coatings. Active coatings aim to improve hemocompatibility solely, so they release substances or antithrombotic agents, and passive coatings provide more like a protective barrier to the bulk material. So they also aim to improve hemocompatibility, or at least not decrease it, but their main goal is to increase wear resistance, which is especially important for areas where physical contact can occur, like between the rotor and the stator. So to prevent scratches, in some areas, high wear resistance or higher than the alloy itself is necessary. So just to quickly introduce active coatings, I'll give you some examples. There are, for example, albumin coatings. That's a protein that kind of lacks platelet binding peptide sequences, so has positive properties. There are anticoagulant surfaces that use, for example, heparin to inhibit the activation of the coagulation cascade. Platelet inhibitor or fibrinolytic coatings exist, and the most physiological approach actually is the endothelialization. So the goal is to form a natural layer of endothelial cells on surfaces by in vitro cell seeding or in vivo self-endothelialization. However, this approach is still very challenging, especially when maintaining a stable adhesion of the coating when the implant is in the application scenario. And talking about passive coatings, which is more the topic that we research on in our lab. When we look again at the titanium alloy, we have a hardness of around 350 Vickers hardness. And coatings, like popular coatings are, for example, titanium nitride and diamond-like carbon, which, as you can see, have increased hardness compared to the bulk substrate of the titanium alloy. Titanium nitride, for example, is already widely used in industrial applications, and diamond-like carbon is even harder than titanium nitride. However, here we have the possibility of micro cracks. That's why multi-layer coatings are necessary here, which I will talk about again on the next slide. So that was basically the wrap up of coating strategies. And now I want to show you what we did in our lab to investigate different coatings of surface properties. So we investigated different hard material coatings as introduced before, and I was talking about the multi-layer coatings. So basically the goal is to prevent these micro cracks. So adhesion layers or bonding agents are applied between the substrate and the top layer coating to provide a gradual change in different material properties, like, for example, the hardness value. So the tension is decreased of the top layer coating, and these micro cracks can be prevented. So we investigated adhesion layers like chrome nitride, and also silicon-doped diamond-like carbon, which also can be applied as top layer coating, actually. And what we also investigated were titanium surfaces with different roughness values on a nanoscale, which we also tried to correlate with the zeta potential. Yeah, so just a quick introduction of our experimental setup. We use a syringe pump and flow chambers, and we perfuse human blood over in the flow channel, over our sample, which is mounted into the flow chamber. And the flow channel is only 200 micrometers thick, so we can create very high shear rates, which are simulated before, so we know the exact scenario. And afterwards, we look at the surface with a fluorescent microscope, and we can visualize the adherent platelets, create a binary image, and then evaluate the covered surface area. So just a quick summary of the results. For the hard material coatings, we could show that all coatings did not increase platelet adhesion, and titanium nitride even showed significantly lower risk for platelet deposition. So according to our studies, we can recommend it for wear-resistant coatings. In terms of bonding agents, it is very important to avoid chrome because it showed high risk for thrombosis. So if scratches still appear on the more wear-resistant coating and the bonding agent is exposed, it is very important to avoid chrome. However, chrome nitride is more suitable since it aligns with titanium in deposition levels. And just a quick overview on the roughness and zeta potential measurements. You can see the covered surface area with platelets in green and the zeta potential in blue. And you can see that there's a generally trend of that suggests that changes in the zeta potential due to nanoscale roughness also influences platelet adhesion, and the lowest mean platelet adhesion for the mirror finished surface here is at the most negative zeta potential, which also correlates with the literature of the negative surface being more beneficial for hemocompatibility, in general. If you're interested in these results in more detail, you can have a look at our publications. And with that, I'm finished with my talk. Thank you very much.
Great. Thank you so much, Isabel. It's very interesting, and I think that there are many questions for the future, I suppose, because nowadays the VAD are blocked as freeze in the technology, and think that in the future there will be an implementation. There are some questions. One is sorry, Felix. Okay, they are already answered. Yes. There are any other question from the audience? Because this one was already answered. If not, if we have a yes, please, Jutta. Thank you.
Good morning. Thank you to all speakers for the fantastic talks. I would have a question to Peter Schlanstein. Peter, you cited some other work, and you increase blood pressure and gas pressure in order to avoid bubble formation in the blood when you increase the gas pressure. Do you know if the other studies did the same thing, or did they only increase the gas pressure?
You mean the study from Dr. Kashefi where they used the solid
exactly.
membranes? I think they just increased the gas pressure. So on this point, you don't have to control. I mean, probably you have to measure the blood pressure, but not control it. Yeah.
I disagree with that because if you just increase the gas pressure and not the blood pressure, then you will have effects like we have in diving, that you have bubble formation in the blood because it cannot, the pressures do not match with each other. So I think that's a dangerous pathway, and that should be clear to all clinicians that they cannot just increase the gas pressure and get better results.
No. Yeah,
just
you're
that as a
totally right.
comment.
Yeah. Okay. If that came across that way, then that was not my intention.
I know.
Thank you, Jutta. If I can, I have to put another point on this because with the hyperbaric therapy, oxygen therapy, you remove totally the nitrogen from the body. Normally in hyperbaric therapy, you stay hours. How many days you can support this kind of treatment? Because we know that the lungs needs nitrogen for keeping the alveolus shape, because it is observed that if you go totally with oxygen, you will observe the recruitment in the zone of the lungs and other mechanism of physiological mechanism are blocked without nitrogen. So my question is, do you have an idea about this treatment for more than hours, or is just tasted for hours?
No, it's because it's not for the lungs. We some kind of build a pressure chamber just for the patient's blood. So the oxygenator is a pressure chamber, and there pure oxygen and no nitrogen is no problem. But we with our device we look more to VV ECMO support, not so much veno arterial, because there you would get a problem with high partial pressures of oxygen if you get it into the brain and something. So we're developing a, let's say, mini VV ECMO support system for patients with acute respiratory failure in danger for NIF failure. And that can run for days and even weeks. So it's ECMO-based technology. So yeah.
Thank you.
Okay.
Thank you. I had a question for Lisa. Lisa, I'm really interested in thinking about the flow thresholds, ECMO flow thresholds, and especially in small babies and neonates and infants. And do you have any updated information on what are the thresholds we should aim for? Your research and understanding of flow in small cannulae and quite slightly bigger circuits as compared to the small babies. How can you tell me more about is there any more information that's available?
I would need to we are working on it. So if you get me, it's going to be slightly different dynamics in there, lower flows, but then of course, with the scales, the dimensions, we still may have similar, stress dynamics appearing. So, but I would prefer to get back to you on it so that when I have a little bit more solid scientific background on the specific, because we haven't looked too much on the neonates or the pediatric population yet, but it's in the making, so we should have it soon, and then I can give a little bit more informed input.
No, great. No, thank you. That's really really helpful. Thank you.
I would also have a question to Lisa. I was really intrigued by your simulations showing that on one hand, if you have a cannula with a lot of side holes, that they mainly drain blood through the side holes in an optimal setting or in geometrically optimal setting. But as soon as you insert that into the body and there is a kind of curved blood vessel, that changes hemodynamics quite a bit. Do you have any idea how we could show that effect in the body, or how clinicians could make use of that knowledge?
The tricky here is like there's no easy solution, right? And it's also what we don't have in the simulations and experiments right now is compliance. So of course, with a compliant vessel, and if we're draining more, we may have other effects. So it's this available fluid to drain in combination with vessel compliance and also the cannula. So I think what I see from some of our simulation is really the flow rate. Like if it was that you would have access to the volume flow coming from the vessel that you're actually draining in, and then you could match that to the fluid that you want to drain, then that would actually be quite good. I know that's a tricky value to get. But it's really that dynamics with the surrounding incoming flow and the draining that is the tricky part here, I think. Or that can actually give some more hands-on info on how to position the cannula, especially, and also knowing the vessel geometry. Because as soon as if you have a narrow constriction, and then you're draining less, that I was saying, than what you have incoming, but at the same time you're blocking the flow, then you will have a blocking or a buildup of pressure, but also stagnating the flow because it has nowhere to go. And there, the balance with what you drain as compared to what's coming in is actually quite important. So there's a lot of dynamics here, and then it's going to vary a little bit on the situation. But I see that there are different cannulation strategies that are optimal for different flow rates, and also on the patient side. Yeah.
Thank you.
Sorry, we have just a couple of minutes, and I don't know if Mick is still with us, but anyway, Mick, you are with us? No problem. But my question is to the panel of the discussants, because Mick show us a very high specialist as I could say, pathway for doing the transport for kids. And we like to say, that also Euro also can help the ECMO practitioner for doing a CV for transport patients, particularly for transport neonate and kids. Maybe Aparna or Nick, someone that can answer. Otherwise, I go on.
So, Mirko, just define that question for me. So, what are the?
Yeah. Do you know, because we have many persons connected, not only from Europe, in Europe, also, or a Euro also that is equal, have something that can help to improve the skills for transport neonate or not? There is a training, particular training or not?
I mean, I know that the Euro also has a transport course for here, how to move children and adults even safely in either primary or secondary mobile ECMO. So I think there are training opportunities. But having said that, there are not that many experienced teams who transport. Even in the UK, there is one team, the Leicester team, who transport children and adults on ECMO. So mobile ECMO is a growing field, and I think there will be an opportunity here for more training and getting more experienced teams, because we all believe still in regionalization or centralization of ECMO services. I hope that answers your question.
Yeah, yes, because I want to underline the educational the educational aims of ELSO and EuroELSO, also for the centers that are not experienced in some topics, and Mika show us a very high specialistic topics. And for example, in my center, we don't do neonatal or pediatric, and I would like to know how to do it. This is a strongly important mission that Bob Bartlett, when found also say, because we are doing research for sure, we are doing a clinical study and based on the registry, but also we are to train and educate as much as possible. Also in different regions, as you mentioned, regionalization should be done, and this is our mission for EuroELSO. I think that I just arrived precisely at 10 quarter. Yeah, in Italy it's 10 quarter. I don't know in the other part of the world, and I leave the stage to the next block.
Thank you, everyone. Wonderful.
Thank you so much to my colleagues and friends, first of all. And I saw Roberto Lorusso already connected. Please, Roberto, thank you so much. Have a good continuation.
Thank you, Mirko. It's now 10:15 in Brussels. Can you hear me? So good morning to those in the same time zone, and hello to those in the different time zones. For those just joining, welcome to the European block of this very interesting 24 hours lasting global summit in honor of Dr. Bartlett. This has been a great initiative of Nick and Elzo. I'm Leen Verkaams, and together with Roberto Lorusso and Peter Sherangomski, I'll be moderating the next session. First, quickly, a few words to introduce myself. I'm a perfusionist / ECMO nurse / ECMO coordinator at University Hospital in Leuven, Belgium. And I've been active in the EuroELSO steering committee as a perfusion liaison since the start of EuroELSO. And I have a very special interest in training and education. I'm very honored to be part of this great initiative, as it is thanks to Dr. Bartlett that perfusionists and engineers have been kept intensively involved in the multidisciplinary ELSO family. Dr. Bartlett greatly appreciated our technical expertise and efforts in optimizing the ECMO circuit and management. I'm also very honored to be virtually seated in the moderators panel with Roberto and Peter, making us a very multidisciplinary team, and I will let them introduce themselves before we start with the lectures. Roberto?
Thank you, Leen. Thank you very much. And it is my really deep pleasure and honor being here to celebrate what Bob gave us. I mean, a tremendous treasure, I would say, of example, of knowledge, of friendship. I can tell you how I have memories with working with Bob to establish EuroELSO when I proposed that many years before, and he was really, of course, instrumental to start the chapters, and we had the privilege to start the first chapter with Euro also, but he was really ahead of times always. So it is really my distinguished pleasure to do that. So I am a cardiac surgeon working at Maastricht University Medical Center. I'm full professor of cardiac surgery and ECMology since many, many years. And thank you for having invited me.
Thank you, Lean. Thank you, Roberto, for introducing the session. My name is Peter Shalangowski. I'm a medical intensivist at the University of Vienna and currently a member of the scientific committee of EuroAlso. To start off the session with the first presentation, it's a pleasure to introduce Nina Buchtiller from my institution in Vienna, Austria. Nina is a specialist in internal medicine and intensive care medicine with a strong belief and trust in evidence-based medicine and a particular passion for coagulopathy and anticoagulation on ECMO. Welcome, Nina.
Hi, everyone. Thank you so much for having me. It's a real honor to be here, and I'm just managing my slides. And I'm very glad to present my heartfelt topic, anticoagulation ECMO. And I'm going to talk about some ideas of future directions where this field could actually lead. And also very glad that Professor Bartlett had a very particular interest in anticoagulation, extracorporeal membrane oxygenation, which he has actually quite recently published a manuscript in with some other rock stars in anticoagulation on ECMO. So I imagine when Professor Bartlett started to do ECMO research and when he pioneered this field, he was most afraid of the dreadful outcome of intracranial hemorrhage in these patients. He used unfractionated heparin as his favorite anticoagulant, and he used the ACT and later on the APTT for his monitoring practices. And when we take a look at the 2020s, what actually our current standards then were, we actually see that during the past 50 years in these particular fields of anticoagulation management and coagulopathy management, actually not a lot has actually changed. So the big question is, what will happen in the next 50 years? And that's what I'm going to talk about a little within the next few minutes. And I hope that this will work.
Das hier ist mein Sektor. Das hier ist das wichtigste Gerät des Küstenwächters. Das Gerät. Überlebensradar.
Hello. Can you hear us? Can you? We are thinking. We are thinking.
Hello? This is the German Coast Guard.
We are thinking. We're thinking.
What are you thinking about?
So this is actually about the question regarding outcomes, whether we are actually speaking the same language, because I truly believe that this is something that we could be better about. This is just an illustration for a very beautiful systematic review from my coagulation friend, Hagen Buscher, who already gave a talk in this beautiful summit. And what he actually illustrated in this figure is that we're just using so many different definitions for major bleeding events. So we are probably currently not speaking the same language. And by 2070, maybe we will have a universal clinical definition, or hopefully we will have that for thromboembolic and for bleeding events, which will probably be derived from big data, having severity thresholds that integrate real-world outcomes and have patient-reported measures as well. When we take a look at anticoagulants in 2070, we will still believe that ECMO provides a risk for thromboembolism. But of course, we know, and we will know that in the future as well, that different embolic directions will have distinct consequences, which may provide a rationale for different anticoagulation targets among different ECMO types. And thus far, no trial, neither prospectively nor retrospectively, has shown the superiority of higher anticoagulation targets. So probably we still haven't reached the lower limit. And that was also what has been highlighted by Professor Bartlett and colleagues in the beautiful review I already presented to you, that probably in the future it will be possible to manage ECMO without any systemic anticoagulation in the near future. And due to technical improvements, we will probably be there very soon. There has been a beautiful publication in 2025 in JEST by Whitney Gannon and colleagues who performed the first randomized controlled trial on actual prophylactic anticoagulation for low-risk patients with VV ECMO, where she has beautifully illustrated that it's on the one hand feasible to perform a multi-center RCT on this topic, and that bleeding rates are indeed lower in patients who are prophylactically anticoagulated only. And maybe for high-risk thromboembolic high-risk thromboembolic patients, we will probably have the possibility to use contact pathway inhibitors in these patients. And what we know from retrospective data from my center, which we have previously published, is that lower factor 12 levels are actually associated with less thromboembolic events without affecting the risk for bleeding. And this probably allows us for the possibility to use contact pathway inhibition, which will lead to a decreased risk of cannula and oxygenator clotting without increasing the bleeding risk. And another very important thing we have to do, especially considering monitoring options, is that we have to aim to better characterize the heterogeneity among different ECMO types. And that's just some experimental data from my research group, where we have used thrombin generation assays in pediatric ECMO samples. And actually the very interesting part from these analyses is that peak thrombin generation suppression does not increase above anti-10A levels targeting above 0.4, which means more anticoagulation above this certain threshold was simply just not associated with an increased efficacy. And what we can learn is that probably we have to learn to take care of the heterogeneity among these different ECMO types and just use novel monitoring assays to better characterize the anticoagulation profile of these patients. So this is kind of my idea and my future perspectives of anticoagulation in ECMO. And I want to thank you very much for having me here. I'm very happy for questions and comments in the Q&A session afterwards. And to further optimize our management of anticoagulation ECMO, I'm happy to present a survey that was created by our EuroELSO working group on anticoagulation and coagulation aspects. And I'm happy if you want to take this one and help us to improve the anticoagulation management of our patients.
Thank you, Nina, for a great presentation. Now we will take questions and discuss at the end of these three talks, and now we pass on to Roberto on the next talk. Thank you, Nina.
Thank you, Peter. We'll proceed with Benjamin Fredriksson, who is a senior consultant and head of the preoperative anesthesiology clinic at the University Hospital in Frankfurt from the Goethe University. He leads the data science group at Frankfurt. His research focuses on the standardization and AI-driven analysis of ECMO therapy, and is currently the chair of the Simulation Artificial Intelligence in the ECLS working group of EuroELSO. Benjamin, the floor is yours. Actually, the computer is yours.
Can you hear me already?
Yes.
Yes, thank you. Okay, thank you for the nice introduction and thanks for having me here. So my talk is also about language. So it's a creative thing from Nina. It's all about language. I'm about to talk about standardization and how this will improve and boost the research. So when we talk about standardization in ECLS, we first have to get a glimpse out of what our machines are doing. You know, you treat all patients in the ICU, you know what these what the ECLS machines are measuring, but taking into account that they measure per variable every second or millisecond data points. If we sum this up for each variable, we get over 86,000 data points per day, and sum up all of those, we got over 1 million data points. But they are all lost right now. Every second we're losing these quite amount of data because we're missing a way to access it. It's quite expensive for ICUs. In general, if you have a patient data management system that's electronic, most of the times it's written per shift with a few variables. So we're losing a lot of data every day, every second. And if we compare it with, for example, the Mimic 4 data set, which is well known, they have a high frequency data set for ICU waveforms, like for ECG, blood pressure waveforms, SpO2. But for ECRs, nothing comparable exists right now. So, and why this matters is when we look and how to incorporate AI into the ECRS therapy and to the bedside, and we look at the patient journey where maybe models could help us improving patient outcome and help us treating patients. As you can see down the data layers, there's the classical clinical context, which is static, but we have high frequency data in nearly all parts from ECMO devices, but still they're not accessible, and therefore the models can't be built out of it. And why this matters even more when we look right now, we don't have any clinical AI models at the bedsides. The clinical reality is that we don't have any. And if we go on the left of the tunnel, it all starts with the data sets. And if we don't have good standardized data sets at the end, and still there is nothing, and there will be nothing coming if we don't have a resolution for the core problem. So the root cause is we have no unified nomenclature. In the ELSO, thank God, we know when we talk each other, we need the same language, and therefore we have the Maastricht Treaty, and we know when we talk about blood flow, pressure, what we're talking about. But the manufacturers and the life cycle of the ECLS devices, they're talking a different language right now when we're looking onto the devices. There are different variable names, and also in the back end on the machine, they're saved in a different way. So when we look to on the second part, the standard data format isn't for every manufacturer the same. When we as researchers or clinicians want to get it out, it's not the same. And also what is really a pain, the structured access to it is quite difficult. It's highly expensive to get access to those data and establishing infrastructure. So in research, we have the FAIR principles, which normally the data should be findable, accessible, interoperable, but all this is not true for ECLS data right now, and therefore multi-center research can't be practically, or is impossible right now. To overcome this, we have to build standard stacks, and we already started in the beginning with the ECLS terminology with the also Maastricht Treaty. But that's the one part, but we need a standardized way, and therefore we have a medical terminology systems like SNOMED CT, but still there is no terminology for ECMO and ECLS devices and how the parameters are tracked there. In a cooperation with Jutta Ahrens, she's a part of the ISO standard group for ECLS therapy. We proposed and addressed these issues right at the ISO and talked with the manufacturers there. And it's on the way that we generate a new ISO standard that the manufacturers are buying, that they have to export in a certain time frame, like the second base, in a machine readable way. So this is proposed to the ISO, and it has to be approved, but still it's not there. Also, the data exchange, we have right now HL7 FHIR, which defines how we get the data out of the machine, but it's, as I said, it's very expensive, and not every device manufacturer is still on that HL7 FHIR format. And luckily, last year, a group also involved in ELSO published from the OMOP Common Data Model, which is also a definition for a data set for research purpose, explicitly a format for ECLS, but now it has to be adopted by the manufacturer, by the researchers, that we can access it. And therefore, we need the data transformation. We need a standardized way, a script, how to get these data into a standardized way. And therefore, in my group, we built these ECMO Reader. It's an open source software where we can easily access the data out of the ECMO machines or the ECLS devices, store it in a structured way with the ECLS CDM system, can put treatment information, patient data entries into it, and get also a data visualization. And with this in mind, the vision should be that we have an ECLS high frequency registry where, because it's highly difficult to get data out of the ICUs, we need a simple data ingestion stepwise project. First thing, maybe a USB upload in the transformation, maybe the ECMO Reader or any other software tool where we can get these data combined in a standardized way to the registry. Or if you're lucky and working in an ICU environment where the PDMS system is already connected to ECLS devices, an automated route from the PDMS to the registry would be good. Of course, if we have a cloud-based system like that, it has to be the privacy by design GDPR compliant. We have to store data which is identifying separate from medical data. And of course, it's a multi-center project. We need the community to support to this, that in future, we have early warning systems by these high frequency data, but also have real world simulation data to learn from complications from patients in a real world scenario. And of course, not a separate registry. We have to get these tools into the ELSA registry to have a broad overview of ECLS therapy. So what can standardized data unlock? It can bring us multi-center research, and we need multi-center research to get real meaningful AI models, because right now, small single center or small multi-center data sets are there, but not granular. We don't have a lot of external validation data sets, and that's really crucial for AI models. And of course, also the regulatory pathway. We have the manufacturers have to face a lot of regulatory pathways. And if we get a registry like this, it would really get the manufacturers more to market surveillance and better device approval. And for us, the clinicians and all the other experts at the bedside, we get the models quite sooner. So the key takeaways is ECLS data is generated in a high frequency, but right now per second, it's lost. We need a standardization to make it accessible. We need the standardization on every level, because otherwise we can't get meaningful data, and without data, no AI models can generate. And like ELSO defines the standards for ECLS care with the Maastricht Treaty, we must define shared data standards to improve the patient safety together, to come from data silos to data-driven ECLS care together. Thanks.
Thank
you
you,
very much.
Benjamin.
Thank you.
Thank you.
Please.
Very interesting. We indeed have a lot of work to do with regards to data and standardization. And there maybe we have to include another discipline, the IT specialists, and then the people who are concerned about data safety. So it's a very complex issue, but thank you. I'm sure there will be questions. But first of all, I'm very honored to introduce our next speaker, Dr. Luigi Camprota. He's a giant in the field of ECMO. He's a consultant in critical care, and now I have to read, and the Joan Rees Chair in Critical Care at Imperial College London. His clinical and academic focus is respiratory failure, and his work centers on the physiology and management of RDS, mechanical ventilation, and extracorporeal support, particularly ECMO for the management of severe respiratory failure. And he's going to give us a talk about mechanical ventilation on ECMO, but only the first 72 hours.
Dean, thank you so much. I hope you can hear me well and you can see my slide. I cannot tell you what an honor it is for me to be part of this 24-hour summit in honor of a great scientist and person, which was Bob Bartlett, a visionary that clearly, as we all know, brought ECMO to life. And I'm just going to go back a little bit to the time and talking about ECMO. And this is the first ECMO trial in severe respiratory failure. And at that time, it's worth looking at the survival curves. And you can see this is the number of patients surviving. And then basically at the end of 68 days, very, very few patients survived. And if you fast forward in 2018, this is the earlier trial, we can see two things which I think are very interesting and part of the topic of this talk, which is the control group. The mortality or the survival of the control group has increased substantially. And this is partly to do with the quality and the type of mechanical ventilation that this patient receive. And when ECMO is added to the mechanical ventilation, and it will allow to do much more to mechanical ventilation, that's when the survival curve will start to separate. And therefore, ECMO is the thing that will allow us to reduce mechanical ventilation. And this follows this idea over time. And I think on the left-hand side is basically what the 1970s were about, which was maintaining PaO2 at all cost. And that was at the expense of what we all know now being mechanical power, so the energy that we give to the lungs. And what we've learned is that clearly as we increase the driving pressure, so the plateau minus the PEEP, which is the force they use to move the lungs over time, and increase the respiratory rate, mortality increases. And so when we look at ECMO, what ECMO will allow to do, and this is the earlier trial that we all know very well, it will allow a reduction in mechanical ventilation, both in terms of tidal volume and driving pressure, and on respiratory rate. And as a consequence, the mechanical power that is delivered to the lung decreases by, on the whole, around 66%. But I want to draw the attention to the respiratory rate, because the respiratory rate goes from 31 breaths per minute down to 23, which is a reduction. But I think we can do some more in terms of improving even further the stress and strain to the lungs. And you can see here, these are the reduction in mechanical power, which is mostly respiratory rate and tidal volume. But I want to show this. This is a new data. They show that the effect of mechanical power by either reducing tidal volume, you can see this is low tidal volume and low respiratory rate, greater reduction in mechanical power. But look what comes next. What comes next is not just low tidal volume, but it is low respiratory rate. So you see the second reduction comes actually from reducing the respiratory rate. And thirdly, having a low tidal volume and high respiratory rate. I'm drawing the attention here to the potential effect of using ECMO to substantially reduce respiratory rate in our population. Now, I've redrawn this from the REST trial, just to show the same point. The same point, this was a trial using low-flow extracorporeal CO2 removal to reduce tidal volume. which clearly the investigators managed to achieve. You can see that in yellow is a reduction in tidal volume. But this was at the expense, you can see that, in the respiratory rate, which increased. And as a consequence, we all know the trial was negative in the sense that there was no reduction in mortality or ventilation-free days. And therefore, I think this is an important point when we think about the goals of mechanical ventilation on extracorporeal support. And this is one thing that, one study that we've just completed, and you can see here the relative effect of tidal volume or respiratory rate on lung efficiency. So this is the amount of ventilation that is required to remove 1 liter of carbon dioxide from the native lung. And you can see one thing, that if you look at patients with the same mean of ventilation, but different tidal volume. So this is tidal volume of 8 mils per kilo, 4 to 8, and 4. So clearly this will have the same mean of ventilation, but different tidal volume and therefore different respiratory rate. You can see that the lung is much more efficient when the tidal volumes are increased and the respiratory rate is as low as possible. And this is another giant of respiratory and extracorporeal support, which was Luciano Gattinoni. That's shown in 1978. Similar idea, that essentially reducing the tidal volume would reduce the FRC and therefore over time decrease compliance. Whereas maintaining the tidal volume with very low respiratory rate would achieve maintenance of the FRC and a significant reduction in mechanical power. And this is shown in this is animal, and you can see that as you increase the respiratory rate, what you will actually increase is the delivered transpulmonary energy per breath. And there is a threshold for lung injury, which in this particular case was 12 joules per minute, which was achieved above 10 breaths per minute. And so what we have done recently, this is a study that we've just completed, is by reducing respiratory rate quite dramatically down to 2 breaths per minute. And you can see that while the driving pressure per breath, these are sigh breaths of 2 per minute, has increased, the mechanical power substantially decreased down to 2.5 joules per minute. And 2.5 joules per minute is the same mechanical energy that we are using today here as we are listening to this talk. So it's a very normal mechanical power, which is tenfold lower than what is used in severe ARDS during mechanical ventilation. This is what is possible to achieve if supported by ECMO. And this is a study again that we've done, and this is the cumulative energy during 72 hours. And you can see that near apneic ventilation can reduce it very dramatically. And what is more important, the compliance does not change, because essentially during inflation, we recruit and maintain recruitment at the expense of a much reduced mechanical energy. So you can see the compliance is the same between the two groups, the inspiratory capacity is the same, and the lung ultrasound score remains the same. And this is regardless of recruitability potential. So when we divided patients into highly recruitable and non-recruitable, the effect on lung volume, inspiratory capacity, and compliance is the same. So indicating that actually having a low tidal volume does not lead to de-recruitment over time. And this is supported also from previous, this is an animal experiment from Salvatore Grasso in Italy, where you can see the distribution of aeration or ventilation using CT scan in animals of very low respiratory rate or normal respiratory rate, which is comforting and supports our clinical data. But what we also showed is there's a significant reduction in inflammatory markers. Now, we have seen a similar thing in our clinical data. And you can see here, this is the CRP change over time, and you can see there is a correlation between cumulative mechanical power, and this is mainly driven by a reduction in respiratory rate and the CRP. And what is interesting is also that this is going back to some of the animal data, showing that using a very low respiratory rate, supported again by the artificial lung, can achieve histological changes. They are very similar to control animals. So essentially, there is much more protection that comes from low respiratory rate than from super low tidal volume. And this is quite important. The next thing about lung protection is, if you look at this network meta-analysis, shows two things which are useful. One is that the only two supports the increased survival or beneficial effect on outcome in addition to lung protective ventilation is VV ECMO, and this is the purpose of this 24 hours, and prone positioning. And so the idea is that maybe putting them together will increase the effect or the protective effect on lung injury. And as you know very well, the data have been conflicting, and it's fair to say that the prone ECMO one has not supported this idea, whereas other studies have been more positive. And perhaps this meta-analysis, the only thing we can say pending further data they are forthcoming, the one thing we can say is that there seems to be a beneficial effect of prone position in ECMO in patients with more severe lung disease or PF ratio, who did not receive prone positioning before ECMO. And this was one of the issues with the prone ECMO study, that you'll remember about 90% of the patients had received prone positioning before extracorporeal support. And as we know also from the non-ECMO literature, that prone positioning, when provided early, is much more effective. So this is my summary slide. And once a patient is on ECMO, clearly we can support all the extracorporeal VO2 and VCO2 that will allow us to maintain the most lung protective ventilation possible. But it's important, I think, to do it in stages. I think the first stage is to achieve what we know being lung protective. So through driving pressure, which is the total strain, and the total plateau pressure, which is the total stress of the lung. And at the beginning, very, very slowly reduce the respiratory rate. We know from animal data that a sudden decrease in respiratory rate or a sudden increase in respiratory rate leads to additional lung injury. So it's important to do it very gradually. And I would say pending some review, and we are doing in the UK a multi-center Romeo trial looking at this, particularly 2 breaths per minute, and adjust the PEEP to maintain lung volume. And this is important, for example, using something like electrical impedance tomography or lung ultrasound or esophageal pressure to maintain that, and consider prone positioning, particularly pending new data. And this is basically what we know, the importance of ECMO facilitated reduction in intensity of mechanical ventilation, not just for ventilation-induced lung injury, but for the downstream effects on multiple organ failure and mortality. And with that, I would like to thank you for the honor of being with you today and celebrating a great person and a great scientist, which was Bob Bartlett. Thank you so much.
Thank you so much for the always great presentation, Luigi. Now, as the next speaker, it's a pleasure to welcome Haifa Lister, who is a consultant pharmacist in cardiothoracic transplantation and mechanical circulatory support at the Royal Brompton Harefield Hospitals. And she's managing the pharmaceutical care of VAD and ECMO patients, as well as heart and lung transplant patients. And her research interests include personalization of dosing in special populations, for example, critically ill patients with or without ECMO or renal replacement therapy. Thank you for being here. Haifa, here's your stage.
Thank you. Can you see my slides? Oh,
Yes, we can.
okay, fantastic. Right. So again, thank you for the invitation and the great honor to actually be able to be part of this Legacy 24-hour Global Summit. So I'm just going to do a quick, and I apologize that some of this is going to be quite basic, but we know that from lots of other things that some drugs are quite sticky and will stick onto various bits of materials such as tubing and the membrane oxygenation. I've put in there things in red are where ECMO may affect the way we handle drugs, and so those are the ones that have been of interest. So the tubing, as well as the membrane oxygenator, because it's a large surface area. And then, as mentioned by previous speakers, a great proportion of patients are also on a hemofiltration unit, which in itself can also affect the way we handle drugs. So why, obviously as a pharmacist, pharmacokinetics I find very interesting, but it is important because if a drug's going to actually have an effect in the body, it needs to be in the right place at the right concentration for the right amount of time. So knowing how a drug's handled will help us guide what dose to give, how often, and whether to adjust it in certain medical conditions, and whether we would expect drug interactions. Currently, a lot of the time we use generic product information that doesn't account for any physiology and PK differences in critically ill patients. So this figure shows us the changes in the PK due to critical illness, as well as those that are additionally ECMO-related PK. So critical care patients are complex with a number of patient and disease effects, which can affect all the absorption, distribution, metabolism, and elimination of drugs. They often increase the volume of distribution and alter the clearance, so the way we would clear the drug, and can affect half-life, which can then also affect the dosing interval that we should be giving these patients. I'm going to focus mainly on treating infections. It remains a significant challenge for intensivists, and the ECMO circuit probably represents another PK compartment because they potentially could sequester some more drugs and increase volume of distribution. I'm going to keep coming back to therapeutic drug monitoring in this group of patients. If it is available in your center, is probably going to be a key component in managing these drugs. So in terms of ECMO, there are three parts. There's understanding the drug properties. So I'm going to see if I can get the laser pointer. So here, in particular, is the degree of protein binding and the lipophilicity. Knowing those will help us understand how that's going to be cleared. The ECMO circuit, and then patient factors. Critically ill patients are complex. As I said, so some of them will have renal, they'll be hyperalbuminemic, they might have inflammatory responses. The inflammation can affect the way, again, we handle drugs in this patient group. And then there's also the thing about VA is non-pulsatile. And, the kidney interprets this as pulseless blood flow. Is there a difference between VA and VV? Adding in renal replacement. And then there's the concept of saturation. Over time, do all the binding sites become saturated? Do you have competitive binding between different drugs? All of these are unknown kind of questions at the moment. And organ dysfunction, like I said, is common in patients receiving ECMO, especially prone to renal injury and decreased drug clearance, as well as liver problems. So drug PK factors are your lipophilicity, which is a thing that we call the log P. The higher the number, the more lipophilic a drug. The degree of protein binding, these in turn affect how spread around the body it is, which is your volume of distribution. The higher volume of distribution means it's not really just held in the blood volume, and then the clearance, whether it's cleared by the kidneys or the liver. And then with anti-infectives is what we call the PK/PD index, and that is, are we looking at getting a peak over the MIC? Is it the time over the dosing interval, or is it the whole exposure? This just gives us a summary of your protein binding, though, and high lipophilic. You can see there are some drugs that are in both camps.
but anti-infectives are a heterogeneous group of drugs. So where does that leave us? Now, these questions started being answered about 10, 11 years ago by groups in Australia, where they did ex vivo experiments looking at the relationship of study drugs, lipophilicity and protein binding with the circuit. And they looked at whether this would have implications on both the choice and the dosing. And they found that recovery of highly protein-bound drugs such as ceftriaxone and caspofungin was significantly lower in the ECMO compartment, ECMO circuit versus the controls. And that for drugs with similar lipophilicity, then it was the extent of protein binding which can determine circuit drug loss. And they advise that future PK studies should focus on drugs with greater lipophilicity and protein binding and to use TDM in the meantime. So that brought us back over at our institution, and we started to look at antifungals. And as you can see, antifungals do fit in either in highly protein.
I'm sorry to interrupt you, but are you showing slides? Because I think, are you seeing the slides? Because it looks that they're not on the screen.
Oh.
I could see them until 5 seconds ago.
Yeah.
Roberto.
But then it disappeared, right?
No, I can see them normally.
You can see, okay, so maybe
try again.
Now it's coming back. Yeah, thank you.
Okay. Okay. Can you see? Yeah, so you can see my slides. Can you see me moving them on? Yes.
Yes.
Okay.
Yes.
Sorry. So then at our institution, we looked at doing ex vivo studies, looking at focusing particularly on these antifungal drugs. And we found that voriconazole, although there was a little bit of a difference, was not significant. The big significance was with posaconazole, which is not unsurprising because it's highly protein bound and highly lipophilic, and with caspofungin, which again is highly protein bound. There was loss in the control as well in the caspofungin, which we think is probably breakdown of esterases in the blood. So then we went on to do a clinical trial, the PK study, of which we're about to publish our caspofungin data. And we're looking again at antifungals, and we're about to add in an arm of amphotericin as we start to use a lot more of that in this group of patients. This was following the ASAP study, again, across Australia, New Zealand, which was a multi-center study looking at a number of antibacterial, antimicrobial drugs and sedative drugs. What they did find that there was marked concentration variability was observed in this patient cohort, and concomitant use of renal replacement therapy further increased these variations. Overall, there was large variations with variability greater than 30% in all PK parameters across all study antimicrobials. So how does this compare to other studies at that time? The PK data of antimicrobials was generally consistent with other studies in critically ill patients with or without ECMO. The ex vivo and in vivo experiments did not adequately simulate metabolic changes in the patients, which probably explained why some of the ex vivo studies didn't translate to then into the clinical studies, and that protein binding lipophilicity may have an additive effect. It did suggest that PK changes on ECMO are more reflective of critical illness rather than ECMO itself, unless it's the drugs that are very lipophilic or protein bound that are sequestered. And that preclinical could be useful, but should be extrapolated with caution. So it is a pragmatic real-world study. Almost half the patients required both concomitant renal replacement therapy and ECMO. There was a small number of patients in some of the drugs, and extended infusions were not considered, of which we know that is more prominent now. And there were nil patients not on ECMO to compare with. So there were a couple of recent review articles, this one from the Mayo Clinic and one from us, looking at antimicrobial PK. And what it does, gives us a bit of a guide to look at the most commonly used antimicrobials with their physicochemical properties, how their PK/PD index is, and then what you would expect, whether they would be sequestered, and with dosing guidelines. And so basically you need to look at the anti-infective drug, the patient factors, and then also the pathogen. In these patients, quite often the MIC is actually increased, and so you might want to use higher doses anyway in these patients because of this increased MIC. And then looking at the ECMO compartment and drug dosing. And basically what it is coming up with personalized dosing. It's not a one size fits all anymore. And as can be seen, this is one of the papers that's came out of the ASAP study looking at caspofungin. And as you can see, it differs between the Candida species, and they found that the licensed caspofungin dosing regimens may not be optimal for critically ill patients receiving ECMO, especially in the early course of therapy, and recommended doses of about 100 milligrams daily would be okay for empirical in caspofungin dosing in susceptible Candida species. But as you can see, Candida parapsilosis is not one that is usually going to be covered, even with higher doses of caspofungin. What about if you add in critical illness plus ECMO plus renal replacement? These are, as you can see here, are not additive effects. And again, we can probably postulate about how these drugs are going to be handled in these with these advanced therapies. But again, being guided by therapeutic drug monitoring would be ideal to guide these patients. So going forward, how could we start to be more clever about how we do some studies? One modeling technique is using physiologically based pharmacokinetic models, where you would build in your drug specific properties from the data when they market the drug, the individual organism, and then the study protocol. And then you basically, as an iterative process, keep fixing or fitting the data. So what this does is you have each compartment in the body, you can add in an ECMO compartment. So again, in theory, you could add in also a renal replacement compartment. And what it means is that you don't have to then potentially repeat all the studies again. You could repeat the ex vivo bits, replace that compartment, and then you would be able to hopefully be able to predict in the next group, because there's, as we know, ECMO and devices are continuously being updated and improved. So I'm part of the ECMO PharmNet group, and we produced a Delphi study looking at pharmacological research agenda, and we came up with 6 priorities, which are the PK/PD reporting, interactions with renal replacement, antimicrobial dosing, as well as then looking at analgesia, sedation, as well as optimization of anticoagulation. So key take-home messages: there is a lack of established dosing protocols for antimicrobials and ECMO-supported patients. It can alter antimicrobial PK, but critical illness itself may be a more significant contributor. Delayed renal dosing adjustments in transient acute kidney injury during the first 48 hours for wide therapeutic index antimicrobials would be preferable. And dosing should be optimized using TDM. And for the beta-lactams, use of extended or continuous infusions, in particular if you have no access to TDM. So look at identify your high-risk drugs, arrange TDM and optimal sampling times, and then document a multidisciplinary plan and reassess continuously. ECMO plus evolving organ dysfunction is always going to have changes in drug exposure from day to day. And then basically build your superhero team to be able to adjust and guide the dosing. Thank you very much.
Thank you very much, Haifa, for your comprehensive presentation. And we can open the question and answer time. And we have no questions in the chat, at least as far as I can see. So I can start with the first question to Luigi Camporota. Luigi, what do you think about the results? I mean, you show clearly that the respiratory rate and the prone positioning were the 2 main factors for benefit and advantages in terms of ECMO related outcomes. But How do you explain the negative results of the PRONECMO study? How do you think, what were the weaknesses of this study?
Well, I think I can see Matthew Schmidt being also there, so he might have an idea. But my thought at that time was that quite clearly sort of was a population, so there was a COVID population, and I think 90%, sort of very large proportion of patients had already received ECMO prone positioning prior to ECMO. So the question is, are we selecting a group of patients who are sort of either less likely to respond to that combination? And then the other question is how we drive the ventilator during ECMO as well. That's during prone positioning in ECMO, which is very, very important. So it'd be very interesting to see the next trial, particularly in patients who have not received ECMO, prone positioning prior to ECMO, and then particularly the combination of a low intensity of mechanical ventilation with a lung volume preserving strategy through PEEP titration in patients who are either responsive to prone position prior to ECMO or they are prone position naive and very, very early post ECMO cannulation. So that would be my thought. We know that from even in the non-ECMO population, there is a profound interaction between the type of mechanical ventilation provided and the prone positioning. So I think there is plenty to do in this field, and that's the way I see it going forward.
Thank you. Lynn?
Yes, thank you for this nice answer. I think we can come back to this, but Nina has to leave, from what I understood. And I have a few questions for Nina. So before you leave, maybe you can answer those questions. Your chat was about the future of anticoagulation and anti-10A, APTT, heparin. That's what we're all doing. That's what we are doing. But there are an increasing amount of data supporting the use of direct thrombin inhibitors. So that's my first question. You think we could gain a little bit more, maybe more in the pediatrics? And then a second one, so that's the focus on thrombin, but you think we should also give an additional focus on what's happening in the circuits, like the platelet aggregation? Is there something that we could gain there also?
Thank you, Lynn. So getting back to your first question about direct thrombin inhibitors. So first of all, I think it would be super cool to have an alternative to unfractionated heparin, just to have an alternative. But thus far, I think we're not there to say that direct thrombin inhibitors are equal or even more effective than unfractionated heparin, although the data kind of suggests that. But I think we have to be very careful with final conclusions, because all of the data we currently have is actually retrospective data with kind of before-after studies, where of course also centers' experiences increase when they've switched to a DTI-based anticoagulation strategy at later points. So I think that's just not fair to compare. And thus far, we've recently published the first pilot RCT on comparing DTIs versus unfractionated heparin. And we have actually seen no difference in clinically relevant endpoints. But anyways, I think it's about to get more experience with direct thrombin inhibitors. But one of the limitations we do see with them is that our monitoring strategies for DTIs are just probably not as good as it is for unfractionated heparin. I think we are already at the point where we can say anti-10A assays, they correlate good with heparin doses, maybe not with clinical endpoints in the end, but anyways, I think this truly reflects heparin doses quite well. And we do not have a true alternative for DTIs at the moment, where most studies actually are based on APTT measurements, where we know that APTT is just like influenced by many different different confounders that are just frequently present in ECMO patients, such as factor 12 deficiency or lupus anticoagulants, if you're using an MPTT assay that is sensitive to lupus anticoagulants. Regarding your second question about platelet inhibition strategies, I think that's actually quite interesting because some years ago, my center actually published a randomized controlled trial on additional PGE1 inhibition in patients on VV ECMO, where we've actually seen that patients with additional PGE1 in addition to low dose unfractionated heparin, they had a reduced rate of bleeding and thromboembolic complications. So our idea from that, or also our rationale why we used this substrate, was that there is some preclinical and in vitro data that PGE1 actually kind of protects platelets, so they will not be activated upon very low threshold triggers. But anyways, if there's like kind of a severe vessel injury, they will still be capable of releasing their granula. So that's the idea of using PGE1, and we've actually seen this in a randomized controlled trial that it was actually benefiting patients receiving PGE1 in addition to low dose unfractionated heparin.
Thank you. That's a very complete and very passionate answer. Thank you.
We have a question for Haifa regarding the impact of different equipments and disposable circuit about drug pharmacokinetics. I mean, do you assume that all these findings, all these studies are, I mean, applicable, the results are applicable to all circuits, or should we check circuit by circuit in terms of type and material, of course? I mean, the coated, uncoated, what do you think? Or what are your answer?
So I think the jury's still out. I think for drugs where we do think that they may be at risk of sequestration, different materials are going to affect the way, to the degree of sequestration. And we know that from the earlier ex vivo studies where, the materials were more silicone-based and different types of membrane oxygenators. So I guess if you're going to extrapolate, is look at studies that have similar material to the ECMO circuits you're using, which is why I was saying about the PBPK modeling. I think that's going to be quite a good space for future studies, because what we would just need to do is rerun the ex vivo experiments and then fit it into the model, and that will help us predict, instead of almost having to redo each like a PK study for every single different type of device. But I think, it is going to change. And as materials get more inert, maybe the risk of sequestration will go down even further.
Thank you very much. Any other question? Well, please, Lynn.
Yeah, if there are no other questions, I would like to ask a question to Luigi. You hear me, Luigi? You're still
Yes,
there?
I can hear you.
now.
Yes, I saw that there was a question online for me as well. Yeah.
Okay.
Oh, here, yes, sure. Please go ahead. Go ahead, Luigi.
Lynn, you go first, and then I'll answer the other one that is online. Yes.
So in your algorithm, I'm a perfusionist. In your algorithm, you focus on everything about mechanical ventilation. But I think the key is also to optimize the oxygen delivery from your circuit, because the more you give there, the less trouble you'll have ventilating your patient in a less aggressive way. So I've heard recently a lot about hyperbaric oxygenation, and the aim is to try to augment the dissolved oxygen to very high levels, PO2s to 600-700. So we always learned that the biggest contribution to oxygen delivery is the oxygen bound to hemoglobin. So do you think there is a difference? There is something that we can add and give extra to our patients by augmenting those dissolved, the load of dissolved oxygen?
Yes, I mean, it was very interesting to hear the presentation earlier on. I'm not if we are talking about oxygen as a way of reducing intensity of mechanical ventilation. I'm not really sure whether, especially in the high flow, there will be an additional advantage. It's probably more in the lower or mid flow. And certainly we have not seen any change in DO2, oxygen delivery by adopting a more conservative mechanical power approach. I think what you're saying is really, really important, particularly in terms of VCO2 of the membrane lung, sort of way of optimizing that the right level of carbon dioxide, particularly during transition from a mandatory ventilation to assisted ventilation, where these patients have incredible drive and their CO2 threshold level and apneic threshold might be changing during their illness. So that I think is a key thing to save sedation, paralysis, and additional lung injury from PCL related injuries. Yeah, so yeah, I don't know whether I've answered your question, but I think it's probably more relevant for the lower flow. Yeah.
Yeah. Okay, I'm anxious to see their results.
Yeah, it's been really interesting. Now, there was one which is very close to my heart, the question that the people have asked about TCAV or APRV TCAV methodology in VV ECMO. I think it's a great idea. We've got very little data at the moment, although we've just I think a paper that we've submitted has just been hopefully accepted using digital twin technology in APRV. And what you can see is essentially will allow to reduce respiratory rate really low, maintaining lung volume. So I haven't got a huge amount of comparative data with a traditional one, but I think the background might be promising. So it's something to test in a clinical setting in the future, but the physiological rationale, I think, is there. Thank you for asking.
I would have the last question because we are almost there to Benjamin. Benjamin, what is your idea regarding an immediate use of AI in ECMO? It's just about data collection. It's just about prognostication. Where is, in your point of view, let's say the closest or the easiest way to apply AI in our daily practice now?
Thank you for the question. I think it's really about the data set, and if we look at the data set, if we would like to have a model which is how to prevent any complication or how to improve outcome, or when weaning should start from ECMO, it's a really difficult situation with a lot of data sets, not only coming from ECMO devices, also coming from the ICU. And I think the first what we will see is maybe only tools or models from the devices, like prediction for thrombosis, clotting of the oxygenator, where we slowly getting information out of the machine, and with standardized data, I think there will be meaningful and truly models out there if we can validate them externally in different settings all over the world, not only in the USA or in Europe, in one country. We need globally data set to get an external validation, to get really trustworthy AI models.
Thank you. Lynn.
Yes, thank you, everybody. Great lectures, great presentations. If anybody has more questions, you can just put them in the chat. I'm sure somebody will answer them. And unfortunately, we have to end this discussion and leave the screen to the next block. So I want to welcome Malaika, Mathieu, and Constanti for as moderators for the next session. Have a nice day.
Thank you all.
Bye.
Thank you.
Thanks.
Thank you, Maleka. So welcome for this last block of presentation. So I will moderate it with Maleka and Konstanty, and maybe we can already start with Alex Supadi. Alex Supadi is an intensivist, interventional cardiologist from the University of Freiburg Medical Center in Germany. He's a senior physician at the Department of Interdisciplinary Medical Intensive Care, which hosts a complete ECMO program, meaning that he's doing VV, VA, and also ECPR. So the floor is yours, Alex. Thank you.
Thank you very much for the introduction and the invitation, first of all, to this very interesting program. It's a very long program, and I didn't have the time to listen to everything. I think I should be able to give my slides. Sorry, I just have to. Now you should be able to see them, right? Could you just confirm?
It's
Now
okay.
we can see them.
It's okay. Put it in full screen, please, and it's okay.
Is it in full screen?
Yeah, it's good. Good.
Okay, perfect. So I was invited to talk about the role of eCPR and expanding eCPR from niche to standard of care. And this is really something more about a question mark behind it, because it's really a question if it's something that we want to or that we already have expanded from niche to standard of care. But approaching this question, I want to ask 4 questions related to eCPR. The first of all is, what is eCPR? And it's a general question, but I will show you that it's probably not so easy to really define eCPR for all the settings we are using it in. Then the second one, why eCPR? Then who qualifies for eCPR, which is about indication, which is very important, I think, because it's the major question about the outcomes after eCPR. And then in the end, if we have enough time, I will also briefly show how we can perform eCPR, and which is very dependent on the different systems. But first of all, what is eCPR? And of course, eCPR is the use of VA ECMO in patients with cardiac arrest. But to really define it really shows what we have to concentrate on when we want to initiate eCPR or VA ECMO during CPR. So we defined it in our recent review. We defined it as eCPR refers to the initiation of VA ECMO during refractory, which is very important, cardiac arrest, to restore adequate blood circulation and tissue oxygenation, which is really what the VA ECMO is doing. And then, and I think this is the most important part of it, when conventional CPR cannot achieve sustained return of spontaneous circulation. And this is so important because we always have to aim for achieving sustained return of spontaneous circulation first. And only if this is not successful, eCPR can play a role and not the other way around. So eCPR is nothing we should do for patients with cardiac arrest without trying to achieve sustained return of spontaneous circulation. And why are we talking about it at all? It's about this. And we have seen all these figures in different versions before, and it's all the different numbers on the left-hand side. So it will be depending on if it's fibrillation or asystole or different rhythms in cardiac arrest, but the effect is all the same. With every minute of CPR duration, survival is going down. And if it's not always starting with 6%, like in this figure, in the end, it all goes down to very low survival numbers after 20 or 25 or 30 minutes, and almost none patient is surviving after more than an hour with conventional CPR. And why is it so? This is so because of this. And this is a study, I like it very much because it's from a cadaver and it really nicely shows what we are doing during cardiac arrest. And what we achieve with conventional CPR is blood flow on a very low level. So we cannot achieve good circulation for the patient, but only about 25 to 40% of limited normal circulation. That's why during cardiac arrest, if we do CPR, we still have the patient deteriorating with every minute during cardiac arrest. And that's why eCPR comes into play. And these are nice numbers from the Minnesota program. And it's the same again, like the first figure that I've shown, that with every minute or every 10 minutes of conventional CPR, survival is declining. And after 45 minutes, almost no patient is surviving with conventional CPR. And this is different when we do eCPR, which really in the end helps to extend time of cardiac rest and conventional and combined with eCPR to the right. And as you can see here now, after 45, 50, or even 60 minutes, we still have survivors which wouldn't have survived after conventional CPR only, because then we don't really have the opportunity to treat the problem, which in these cases most often is myocardial infarction. And we really have to gain time to have perfusion to treat the underlying cause of cardiac arrest. And this is what eCPR in combination with successful and high quality conventional CPR can help us. And the same has been shown in a different kind of picture from the Prague-OCA study, where we can see that during 45 to 50 minutes, which is the dark orange and dark blue patients, still survive in both groups, in the eCPR group. So if they are randomized to receive eCPR, or if they are randomized not to receive eCPR in the Prague-OCA trial, they survive without eCPR in the early times. But after 45 to 50 minutes, it's only the light orange and light blue patients who survive, and it's only those who have been receiving eCPR. So after 45 to 60 minutes, we really have to find either that we get to ROS with conventional CPR, or we have to go for eCPR to really achieve survival for the patients. Otherwise, it's almost impossible to survive cardiac arrest. And then it's the question, who qualifies for eCPR? Because as I've said, we cannot do it for everyone. It's a high resource dependent procedure. So we really have to find the right patients to benefit from eCPR. And refractory cardiac arrest alone, if the patient is dead for 2 hours already and no CPR has been performed, is definitely nothing for eCPR. And taking all the data that is available, and the data is really not that clear that we have clear criteria for or against eCPR. So especially in the between, there are situations for patients where it's very difficult to decide for or against eCPR. And what we decided to make it more practicable for everyday work, we define pro, con, and stop criteria, which means stop criteria, it's obvious that eCPR cannot help the patient to survive, so we shouldn't do eCPR in these patients. It's frail elderly people and patients who don't want to have eCPR, for example, or extended no-flow times over 10 minutes. And of course, we have also situations where it's very likely that the patient will benefit from eCPR if the patient has received effective bystander CPR and effective CPR measures until eCPR can be initiated. So this is the green part of the slide, where it's very easy, or should be easier, to decide for eCPR. What's making it very difficult is the area in between. There's the con criteria. And because this is not a score, this is only something that can help us make the decision for or against by just looking at the different parameters, looking at the age and the combination of factors that are always pro and con, and hopefully no stop criteria. But it's in the end, it's the mixture of pro and con criteria that may help us to come, and this is the important part of it, to a team decision for or against eCPR in this individual patient. So this is how we perform it, and I think it's a very good tool that can help us in specific situations to decide how to proceed with an individual patient. And how should it be performed? Of course, I think we can be very brief of it. It's VA ECMO, as we always do VA ECMO. So we have a venous line, arterial line, and we drain the blood from the body through the vein and give it back after the oxygenator, after pump and oxygenator back into the patient. And what is important for eCPR is really the setting, how we install or implant eCPR. And in most settings where eCPR is performed in a high quality level, we have two different teams. It's the green team, which is the team taking care of the conventional CPR measures, which have to be continued during initiation of eCPR. And then we have this blue team, the eCPR team, which is quite independent from the green team. So the team leader of both teams are communicating with each other, but in the end, there are two independent teams performing their measures independently to achieve the common goal of return of, in this case, VA ECMO circulation then. And the advantage of this is that it works in different settings. So we can do it on the streets, we can do it in the ER, we can do it in partner hospitals where we come to the ER and have a rendezvous system, for example, in an initiation hospital, which is similar to the system that is performed in Minnesota or in other settings. So the idea is that we always do the same. We have the same teams performing high-quality CPR and high-quality eCPR, no matter where you are, how the situation is. And the question is, what setting or what concept is best for the individual circumstances? And these both figures together nicely show that we have three different settings. We can do it on scene, on the street. We can do it in the eCPR hospital, and something in between, like doing it in an initiation hospital in between. And my suggestion is to do something dependent on the way, the distance away from your eCPR center. If you're close to the eCPR center, you should load and go the patient, do it in the eCPR center. If you are somewhere in between, long distance travel or too long distance traveling to come to the ER, you should do it on site on the streets. And if you are even further away, initiation hospital meeting, like a rendezvous system, might be the best to initiate eCPR. This is what we initiated in our setting with all the different transport mechanisms, so we can do it on the streets, in the hospital, and also in the partner hospital. And I think coming into this system and this setting, this is really something that could work in different individual settings where you can always decide how can you set up your system for your respective circumstances, which is always dependent on your specific local situation. You cannot give a general opinion or general way how you can perform it in every setting, but it's always setting specific and depending on your local circumstances. Thank you very much.
Thank you, Alex, for this super talk and super overview for eCPR. We will, as in the other sessions, take the questions and at a later point. And it's my pleasure to introduce our next speaker, Massimiliano Polastri, who is a physiotherapist in Bologna, and he has a passion for rehabilitation on ECMO, and will talk about that for us today. Massimiliano, welcome. Thank you for joining us today.
Hi, thank you. Good morning, everyone. Thank you for inviting me. And I should share the screen. Could you see?
Not yet.
Oh, just a moment. Okay. And could you see now?
Now we see it. You have to click on full view. Yes.
Is okay?
I think it's okay.
Okay, perfect. So when speaking about rehabilitation on ECMO, it refers substantially to early progressive mobilization and physiotherapy while patient is on support. Rehabilitation includes generally PROM and AROM exercises and motor activities such as sitting, standing, and ambulation in eligible patients, and respiratory physiotherapy as well. Primary goals of rehabilitation on ECMO are preserving muscular mass and function, preventing the onset of ICU-acquired weakness, which is quite common in critical settings and in critical patients, maintaining functional mobility, supporting ventilator weaning and ECMO liberation, and improve and reducing delirium and improve neurocognitive outcomes. So it should be highlighted that the first manuscript, the first work which was published on 2017, released by the ECMO network. This paper was the first that highlighted and provided a practical approach and described in detail the practical step to be done when rehabilitating patient on ECMO. And two primary criteria emerged from the paper, and the first was the use of the RASS as a cutoff score for progressing or regressing in rehabilitation, and the concept of intensity progression for exercise with continuous monitoring of the patient's clinical condition as well. But today, and more recently, on 2025, ELSO has released a guideline for the rehabilitation and mobilization of patients on extracorporeal membrane oxygenation, and this paper substantially provides a very useful algorithm which provide criteria for progressing or regressing during the journey, during the rehabilitative journey of the patient. And the algorithm is substantially constituted by 5 steps with increasing difficulties and detailed actions to be implemented to progressing or regressing toward the journey. This is a very useful algorithm and should be. I like to present this algorithm because it's very useful and could also help those who were not very confident with ECMO. And this is very detailed table. As well, it's very important, and this is highlighted in the ELSO guideline, to provide a stop protocol when a team member should be responsible for interrupt in any circumstances the treatment, and in case of clinical instability or circuit malfunction on patient intolerance to movement. So this is another key point, the possibility to interrupt the treatment at any time and activated by any member of a multidisciplinary team. Other than the guideline, we conducted recently in 2024 an international survey in order to better understand how physiotherapy is really delivered in the real world. So we launched an international survey, and we obtained answers from 29 centers from 14 countries. And we asked about several topics, but I have extracted some of this and to provide here. One, for example, was if yearly physiotherapy was really provided, and yearly physiotherapy in patient with RAS -4, -3, or -2, or namely deeply, moderately, or lightly sedated, which is at the beginning of the journey. And emerged that the majority of centers provided this yearly physiotherapy within the first 44 hours, and this is quite encouraging as a data. And which kind of physiotherapy was provided in this class of patient was compliant with the literature, and PROM, namely passive range of motion exercise and positioning and splinting of the ankle, were the most represented answers. At the same time, we asked about how many times a week physiotherapy was available, and emerged that in the majority of centers, settings 5-on-7 was the most present, although 7-on-7 full service was also present in 25% of the center we contacted. But the information we would like to stress is when patients are on RAS 0 or +1, just a moment, please. When are on class 0 +1, can I interrupt a moment? Apologize. And just a moment, please. Could you hear me?
Yes, we can hear you.
Yes, we can hear you.
Okay, just a moment. Okay. And I go further. It was quite interrupted. I'm sorry for that. And okay, when we speak about which kind of activity is provided to patient on RAS 0 or +1, which is the class more likely to get improvement on rehabilitation, EROM, PROM, and postural passages were the most represented answer, as well transfer to a chair was achieved as an activity. And this is important because RAS 0 or +1 is the class of patient more suitable for rehabilitation and more likely to get improvement from rehabilitation. And again, on respiratory physiotherapy, on patient on class -4, -3, and -2, usually chest physiotherapy is now suctioning were the most represented answer, as well as in bed positionings. And here another piece of interesting information. And after how many hours respiratory therapy commence in sedated patients, and even here we have an answer that less than 12 hours is a usual approach, and even that nasopharyngeal or oropharyngeal suctioning, deep breathing exercises, and manual technique are the most represented respiratory technique on this class of patients. And even on those on RAS -1 or 0, 0 is the best class for approaching a patient, continues to perform manual technique mostly. So this is quite a small picture of from the real world, and could be merged with the ELSO guideline, and this return to us what really happens in the clinical world. So, but it is quite clear that patients who begin early mobilization, and we saw that most centers initiated the early mobilization within the first 34 hours. So those who initiate within the first 72 hours of ECMO initiation are more likely to achieve greater mobility index by day 7. Mobility that should be scored by using the ICU mobility scale, which is a very simple scale ranging between 0 to 10, where 10 is walking independently. And this scale is so constituted like 10 items who provide and describe different kind of activities and different levels of activities. And it emerged that those patients with the lower SOFA category are very likely to gain greater mobility, as well as those patients on RAS 0 or -1 are those who best perform with the ICU mobility scale. So this is another interesting piece of information from this research. In synthesis, the criteria supporting mobilization are hemodynamic stability, stable ECMO, secure cannula position, adequate oxygenation, and absolutely cooperative or lightly sedated patient. At the same time, active bleeding, severe hypoxemia, unstable arrhythmia, cannula issues could be a matter of red flags or contraindication. I would present here an interesting research where this group are enrolling actually adult volunteers, so not patients, but they are preparing this frame which is very particular because could host all the staff for ECMO and could guarantee both good safe standing and safe ambulation for patients. So this could be considered as an emerging future. At the same time, even the use of tilting beds is an emerging tilting beds like this one. This is an emerging, this is an emerging line of intervention, and tilting bed could be useful where, for example, there is a staff constraint or there is there are suboptimal conditions to provide rehabilitation, because these beds provide gravity, and so patients are dealing with anti-gravity activities. And here is an algorithm to decide the use of this bed, and the criteria is, so this group started with a maximum of 18 degrees of tilt as a trial to progress versus a more higher inclination. I would also draw your attention to this communication, which is quite interesting. And this group has analyzed the video which depict patients walking on ECMO, and they have observed and they have searched in the net for this video, and they extracted for some useful information. And I would encourage you to read this paper, but I have just extracted some topic, and it emerged that the number of professionals involved in the mobilization ranged between 3 to 8, and patients were even supported with walkers, and safety was guaranteed by following the patient with the CER. So I would like to remember you the previous slide where the frame, when I show the frame. So I would encourage you to read this one. Ambulation is often stressed in the literature as a main outcome. Actually, ambulation during ECMO should be not framed as the main objective, is rather the natural outcome of a structured, high-quality rehabilitation program in eligible patients. Rehabilitation even has a role in an economic sphere. Here, some calculation has been done about the impact of rehabilitation on patients who participated or not participated in rehabilitation before lung transplantation while on ECMO. It emerged that those who participated in rehabilitation before lung transplantation during ECMO had lower expenses in several fields, and mechanical ventilation, which is probably the only clinical outmark that we should consider, was substantially different in rehabilitated ambulatory ECMO group, 2 versus 29 days. And having said that, we should conclude that the rehabilitation on ECMO is safe, feasible, and beneficial when performed by an experienced team, and this is a crucial criterion for approaching rehabilitation. So only an experienced team could provide safe rehabilitation. Early mobilization has emerged clearly from the literature, is now the standard of care, influenced, of course, by the cannulation strategy, and the goal is just not survival, but functional recovery. Future direction for future lines of research could be certainly deeply scoring about ADL, activity of daily living, and quality of life on patients surviving ECMO. So this is a piece of information which is missing from the literature. And at the same time, it should be deepened the importance of assistive technologies, such as the use of tilting beds and the walking frames and all the stuff that could help rehabilitation professionals to provide their service to this kind of patients. So thank you so much, and sorry for the interruption.
Thank you very much, Massimiliano. It was very, very interesting and insightful talk. And now it's my great pleasure to invite our next speaker, who is Sebastian Janssen. He's a doctor engineer at the Cardiovascular Engineering Department at Aachen University. So Sebastian, the floor and actually the screen is yours.
Good day from Aachen, and congratulations on the organization of such a wonderful event. Let me shortly share my screen. Okay, so you'll see the presentation?
Yes, we do.
Okay, so thank you once again. I'd like to present two studies that we performed together with our co-workers Franziska Schubert, Jan Heyer, Felix Hund, and together with our neonatologist Mark Schober. And as the title implies, our motivation is to have designated oxygenators for artificial placenta applications. And artificial placenta applications, as you might know, is like the ECMO, a simplified view of an ECMO that is connected to the umbilical cord for extremely preterm infants. We are talking about a gestational age of the 24th week, so we are at the border of viability. And the motivation of these techniques is to bridge the lungs to maturity, or to provide the oxygen transfer via this ECMO system and not necessarily via mechanical ventilation, which is performed now and which is associated with very high mortalities and morbidities in these patient cohort. So the problem from us as a designer, and I'm an engineer, the problem for us is that we have in this patient cohort a very, very low acceptable priming volume. But if we aim, for example, for a therapy goal of up to 4 weeks, we have a growing oxygen demand in this area because the patients simply grow. So this is what we have identified as the requirements for such an oxygenator. And you can see we have a very low acceptable extracorporate blood volume for these patient cohorts. So the complete ECLS circuits, including cannulas, tubing, and so on, shall not exceed the priming volume of 80 milliliters, which is growing a little bit up to the 28th week. And also, like the pressure drop that is acceptable for these patient cohort is very low compared to ECMOs and other patient cohorts. So that is if we want to get rid of the blood pump for the ECLS. So if you just want pure passive perfusion through the oxygenator, we want to be in this area of pressure drop even lower, which would be favorable. So the requirements or the challenge for us is how can we achieve an oxygenator that has a very, very low initial priming volume at the beginning of the therapy, but is then able to compensate for the growing oxygen demand later. And the approach that we have investigated are oxygenators with 2 chambers. So at first only one chamber is perfused, and then if the oxygen demand increases, we can activate a switching mechanism, and a second gas transfer active oxygen chamber is added to the first one, increasing the gas transfer performance, and this without interrupting the operation, without deconnecting the oxygenator. Now I need to make a quick digression because, as you might know, there is in general 2 types of oxygenator designs. One is with a fiber mats wound around the core that leads to these cylindrical oxygenators, and the other ones are where the fiber mats are stacked on top to each other. That leads to these square oxygenators that you are also very familiar with. So in order to develop an oxygenator that has 2 different chambers, we had both approaches. The first concept covers an oxygenator, two-chamber oxygenator with a rounded fiber mat. And the principle is this. So we have a core onto which the first fiber mat bundle is wound. And then we have a switching mechanism with different openings. You can see them here. And then we have the second chamber with another fiber bundle wound around this housing here. So the principle is that at first we will have the blood perfusing only in the outer chamber. And then if we need an oxygen demand, if we need to compensate for growing oxygen demand, we can activate the switching mechanism and aligning these openings to open up the second chamber. And we have the blood perfused also in the second chamber. We built a proof of concept and made a feasibility study. And you see we made an in vitro gas transfer measurement where we were able to increase the blood towards physiological saturation values. However, the oxygen transfer rate that we achieved in this first proof of principle are not yet meeting the targets that we have set ourselves. However, you can see that switching to the second chamber increased the performance of the oxygenator in terms of gas transfer. Another matter with this kind of oxygenating was a pressure drop. So we have seen that we can have a maximum pressure drop of 30 millimeters of mercury in this patient cohort. That was so for all the ECLS systems. So our target for the oxygenator was to stay below 20 millimeters of mercury, which is not possible in this first proof of principle up to like the physiological flow range that we want to use. But these were mainly design issues. So we're working on a second iteration, and I cannot show the results yet because they're preliminary, but they look very good that we can fulfill the requirements with the second design iteration. So we have excellent priming volumes, yet not sufficient gas transfer in this concept. We have a too high pressure drop, but as I was mentioning, we have a second iteration underway. The second concept is a two-chamber oxygenator using stacked fiber bundles. And for this, I need to make another short digression because in order to seal the open fiber ends of the stacked fiber mats, we need to make a potting, a sealing, and we use centrifugal forces in order to create the seal. So if we want to have this as a result, where we have two boundaries made from the potting materials, but with interspace in between, so that we have two compartments actually, we need to find a novel manufacturing potting technology. So we use this first one, which is pretty much conventional round potting to create the outer layer, and then we induce fluid that acts as a barrier for the second potting. So the second potting is then initiated here and hardens. And after the second potting ring is hardened, we can remove the fluid and we can remove it completely so that there are no remains on the fiber bundle. And this results finally in the creation of two compartments within a stacked fiber bundle as well. So this is what it's looked like. In reality, again, for this patient cohorts with very, very small priming volumes, in each compartment. And we also performed the proof of principle experiment in this one. The switching mechanism you can see here. So in this case, at first only the inner compartment is perfused with blood, but then if we act on the switching mechanism, it translates into a linear motion that opens up the second chamber over here, and so the blood can now perfuse both chambers at the same time. This is our proof of principle, and we can see here nicely that only the inner compartment is perfused by blood. Here you can see part of the fiber bundle from the outer compartment, and if we act on the switching mechanism, we can see that both compartments are now perfused by blood. The oxygen transfer was successful in this proof of principle study. This is our target area with the variant. We are very close to missing the target area in the single compartment operation, but when we switch to the dual compartment operation, you see that we significantly add gas transfer performance and are above our target area for this patient population. Again, here is also a second iteration on the way, because what we did not achieve with this one was carbon dioxide removal. Also something with the design issues. We had a crushed fiber bundle that prevented this. So this is also solved in the second iteration. To be of note, we had an excellent pressure drop. So when we measured the pressure drop here over the complete flow range, we did not exceed 1 millimeter of mercury. So there was an advantage of using the stacked fiber oxygenators. So we were very happy with that. But again, a second iteration is to be published. As take-home message, we find that multiple chamber oxygenation or two chamber oxygenation, and there's no limit. We can also have multiple chamber oxygenators, that they are feasible. We still need to do a little fine-tuning to have oxygenator modules that can fit this specific patient cohort. But we also have applications or might have applications in other fields, because we can either switch to add an exchange area, but we could also switch from one compartment to the other in order to add fresh fibers, to have a fresh fiber bundle without ever changing the oxygenator itself. So there might be other applications than artificial placenta applications as well. With this, thank you very much, and I'm happy to answer any questions.
Yes, thank you. Thank you very much. And now I think it's time to move to a discussion. Do we have any questions from the audience?
Thank you so much.
Yeah, Jutta.
Thank you to all speakers. Sebastian, I would have a question about your growing oxygenator. Do you know why, if you increase the gas exchange area by 100%, why you don't increase the gas exchange by 100%?
Yes, thank you, Jutta. Yeah, we were wondering of this as well, and it was design issues. So we identified, in the Bond oxygenator, we had a fiber bundle that was not a double fiber mat where we have the orientation of the fibers like this, but it was only one orientation. So we suspected that we have an unhomogeneous distribution of the fiber density, and thus shunt flow in one of the compartments, which leads to the discrepancy. So one of the compartments was not performing to its full capacity. And we had a similar case in the stacked fiber bundle as well, where we use one of the potting rings as a sealing for the switching mechanism, and this sealing was a little bit compressed. So it compressed the complete fiber bundle and also led to different industrials there. But we solved these in the second iterations.
Then I would have if I may, a second question quickly to the ambulation topic. You nicely showed our work on the ECMO system. Thank you for citing us there. We have issues finding a company who would be willing to really bring that device to the market. And there's a question mainly to all clinicians. We are thinking about making that an open access design. Would that be a solution for hospitals to make use of the ECMO and build that basically with our plans yourself?
Can I answer?
Yeah.
Okay. So the frame we considered in the presentation was like something which is patenting, and I guess this is the future. A name. It should be it should be developed something safe, something available, and I am not really sure that out of market or something like open is a good idea. Rather, something more structured or something more from a professional line could be more helpful, even with the participation of clinicians. But the production and the projection of some of frames or ambulation heights should be deserved in a professional arena, perhaps.
Yeah, I mean, that would be our preferred solution, but so far we didn't find a company who would be willing to take over.
This is quite difficult, probably, because companies maybe are looking for something which should be sold in a large scale, in a large volume. And we know that ECMO is like a niche, is like a narrowed field. And probably this is the main gaps that prevents company to invest in such a kind of products. But the future is certainly in this direction, namely in enhancing and expediting the patient journey with safety first at all times.
Sure.
Mathieu?
Thank you. Yeah, thank you all. First, thank you all for your very nice presentation. I have a question for Alex. Alex, can you say a few words about hypothermia? Because there are some specificity in terms of patient selection. And do you have any tips or advices to convey and to provide to the audience about specifically ECPR on hypothermia?
Yeah, it's a very good question, but very difficult to answer because hypothermia is not hypothermia. And it really depends on the way the patient got hypothermic. So any patient in the end will be hypothermic after cardiac arrest at some point in time. So if you have a patient with asystole that is hypothermic, may just be dead. So this is the first thing. But we have just had a patient, a very young patient, a psychotic patient who went swimming now in February or January, and was hypothermic and got cardiac arrest and got CPR for, in the end, it was almost 2 hours until eCPR, and the outcome was perfect. So this is really the patients that may benefit even after extended periods of time. But patients like avalanche victims who get hypoxic first and then hypothermic, this is a different set of patients and they don't have a good outcome. So hypothermia, as we know, is very preventive for brain injury. So if you like a patient that gets hypothermic after drowning or swimming first hypothermic and then is drowning, this is a good chance for survival. Avalanche victims is a smaller chance because of hypoxia. So this makes it so difficult that you cannot just make it dependent on the body temperature or something. You really need to have more information about the origin of cardiac arrest and the way the patient got into cardiac arrest. And another question, maybe I can add to that, is the question about TTM after eCPR. So this is another open question. We know that the data is now getting worse and worse for suggesting TTM after cardiac arrest in general. But I'm very cautious about that because eCPR patients are a different cohort of patients. They have prolonged cardiac arrest, prolonged resuscitation periods. So we in our center at least still cool down the patients in general on 33 degrees after eCPR. But it's a different part, of course, of the question.
Thank you. Malaika?
Yeah, Alex, thank you all for the very interesting talks. I have 2 questions first to finish this topic with Alex. We do the same after eCPR. We cool our children, and then Sebastian, I'm the only pediatric internist in this moderation group, so I'll have some questions for you afterwards. But Alex, we do cool our patients too after ECPR. And you said you cool them to 33 degrees. For how long and how fast do you wake them up in practice?
So we cool them 33 degrees for 24 hours as fast as possible. So we definitely want to achieve it as fast as possible, which is easier with eCPR than it's after conventional CPR, of course, with the extracorporeal circulation. And so we maintain it only for 24 hours, and then we raise the temperature by 0.2 degrees per hour. In some patients, for example, patients with bleeding complications, of course, we also have patients that we don't cool down for 36 degrees, and then it's easier to wake them up if possible, so we don't have to wait for the body temperature to come up again, and we would wake them up as early as possible. But of course, the patients who have been cooled down won't be woken up with 33 degrees. We wait until 35.5 or something to try to wake them up. So it will be another 24 hours in the end. At least 20, it will be, in general, will be 48 hours. And normally, in most ECPR patients, we just stick to the normal procedure with evaluation after 72 hours. Yeah. We deviate only from this if we have very bad signs early on. Of course, then we may stop treatment early on, but it's unlikely. If the selection for ECPR is well, we at least should wait until the 72 hours if we have clear signs of bad outcome before we stop the treatment.
Thank you. And Sebastian, a question to you now with these low flow mini oxygenators. As I'm a clinician, I found it very interesting what technology can do, but I have a practical question. Do you see like complications, fibrin, thrombos, and how long does one of the oxygenators last? Because, you know, in practice, after 2 weeks, maximum, 15, 20 days, you need to change the oxygenator often in real life later. And you just say the child is growing from 24 to 28 weeks, makes it 4 weeks, and you just extend the chamber. So there I'm nosy how that works in practice.
That is a very good question. Thank you very much. And you can imagine that, like for artificial placenta technologies, there are a lot of challenges that needs to be addressed. And this is only one very, very, very small excerpt out of this. Indeed, like the long-term stability of the oxygenator is one of the unresolved questions so far, especially in this patient group where we need to take very much care of the anticoagulation treatment due to germinal matrix bleeding and stuff like that. So this is going to be a huge, huge challenge. So there are different approaches. Like, I mean, there's the approach, Jutta, correct me if I say something wrong, but you have with the art plug, you have the approach of providing oxygen transfer with the oxygenator over a period of 2 weeks and then switching to CPAP in order to through the rest of the therapy. We have the aim to maintain as long as possible on the oxygenator. We will have to see like further preclinical and in vivo data on the long-term stability of this oxygenator. But absolutely, I absolutely agree. This is a problem and a challenge that we still need to address, like in every other oxygenator. But that is where, like with my last message, where I think that maybe we can also use the different chambers to switch from one chamber to the other in order to have fresh, unused fibers, unclotted fibers. In the case of like when the first compartment is failing, you can switch without ever changing the oxygenator itself, but you can switch during operation to a fresh compartment of fibers. So maybe there are also applications in this case, but right now we don't know how long this oxygenator will perform.
But what works in our favor, because we are working in the same field, artificial placenta, is that we are planning to not use a pump. and the pumps are the main course of microemboli in the oxygenators. So I think that at least that works in our favor and makes the whole system more hemocompatible and probably longer lasting than the typical ECMO circuits with a pump in there.
Thank you, Jutta. Excellent.
Konstanty, do you have a last question?
Okay, I have a question to Alex also regarding cost effectiveness of ECPR. What is your estimate regarding to the proportion of cardiac arrest victims that really qualify for ECPR? Because according to my calculations, it was approximately 5% of people in Prague.
That's a very good question. Definitely, it's a small cohort of patients. However, no matter how you calculate it, it's a small cohort, and this is what all the data and all the simulations show, that a small amount of patients will qualify. And the question, I think, is probably something from the other side. The problem is that a large amount of in the overall cohort, there will be a lot of patients that don't get access to ECPR because they live in an area where ECPR cannot easily be provided. That's why I think it is important to think about, like my last slide, to think about networks and the provision of ECPR with the highest standard, like what they, for example, now plan in Minnesota, how you can reach out from the centers, from the experience centers to a broader area to include all the patients in need, because no matter what you do, it will be a small cohort, and the small cohort will be distributed in a wide area if you're not only focusing on metropolitan areas. And this is, in the end, not only a financial question, but an ethical question. Is it ethically advisable or acceptable to provide ECPR as something we believe that is working only in metropolitan areas, or what do we have to do to reach out in other areas, still maintaining the quality that is required and the experience that is required to provide high quality ECPR for this small amount of patients? And I agree, it will be something like 5 or even less % of the CPR population.
Right. Thank you.
Thank you very much. I think we have maybe 1 or 2 minutes left. So I have one last question before Massimiliano. Massimiliano, the team study, the study that was published in New England Journal of Medicine a few years ago, which was investigated early active mobilization in patients on mechanical ventilation, was surprisingly failed to show any benefit, meaning that it was not associated with the reduction of the mechanical ventilation duration. It was even associated with more adverse events. Do you think that it may, obviously it was not ECMO patient, but do you think that it may also apply to ECMO patients? And probably some cautions are needed in that field in terms of the timing to start early active mobilization.
Yes, I agree in the sense that this should be and could be applied even to ECMO patient, in the sense that ambulation, specifically ambulation, is an activity, is a motor activity that not always overcome the risk to have some adverse event. So which is the advantage to have some meter walked versus a potential adverse event? There is not a clearly advantage. This is because robust, intense rehabilitation should be implemented only in selected individuals who show who are particularly accustomed to movement, who are particularly accustomed to exercise, and who have specific characteristics, namely clinical stability, and even the willing to perform, because patients should be the first actor and should be willing and should manifest the intention, even physically, to perform during rehabilitation. There are, for example, the works of Christiane Perme with left ventricular assist devices. She was a pioneer in mobilizing patients in cardiac assistance. But even in that case, this activity was deserved to select the individuals. So it should not be taken as a standard of care, even if is increasingly performed and is increasingly provided in critical setting. But so my thought is ambulation specifically should be deserved to select the individuals, and even the right time to initiate physiotherapy, namely early physiotherapy, is different in the sense that early physiotherapy should be implemented and could be implemented without particular restriction, because it could consider even passive movement. It could be performed with in-bed activities. So in this sense, early is advised. Early is a strongly advised. Initiate as soon as possible is certainly a good approach for patient on support. But for more articulated activity, so the time should be considered carefully. This is my opinion, of course.
I do agree. Thank you.
Yeah.
Thank you for the question. Thank you very much for this. Thank you all for this very nice session. I think it's time for us, perfectly on time, to let Nick Barrett taking over for the conclusion of this European morning.
Thank you very much, Matthew, Malika, and Konstanty. It falls to me and Christine and Peter to close down the session. So I think the first thing to say is it's been absolutely fascinating. We've had a wonderful journey around the globe, starting in the US, moving to Latin America, then New Zealand, throughout the Asia Pacific region, India, the Middle East, and then South Africa, of course, and then to back to Europe. I hope that you've all enjoyed the bits that you could listen to. This was the first time we've had a 24-hour continuous Congress, and it's been a great privilege to be part of that. We've seen everything from the history through to current practice, through to future practice and some of the advances we're expecting. We've run the full gamut from neonates to small children, large children, adults, kidults, as many of us seem to be, respiratory through to cardiac to ECPR, and all of the ancillary management, thinking about how we ventilate, thinking about how we provide drugs, physiotherapy, and how we generally manage our patients. And it's been wonderful to see a focus on the future and thinking about the engineering clinical interface. So I think that we've probably gone some way to extolling the legacy that Bob Bartlett left us, and to thinking about things in the way that he has thought about things over the years, by being hopefully inclusive and future thinking. So my personal thank you, first of all, to all of you for attending, to all of our speakers with absolutely fabulous talks on such a wide range of topics. The ELSO team with Christine, Alex, Lauren, Peter, and all the technical support in the background. To run this without major snafus is absolutely remarkable. And of course, to my co-chairs, John Fraser, Ibrahim Fauzi, Drigo Diaz, and Peter Alexander, for organizing such a wonderful Congress. So thank you all. and goodbye from me. Christine.
Thank you all again. I think one thing that stuck out for me was the particular attention some of you spent time on talking about Bob and your personal relationship with him, but also how it's affected your own career and your own goals. And I'm hopeful that this Congress and others, as we go through the chapters this year, will continue to let his legacy live on for a very long time through us all. So thank you so much for the authenticity, and the future is bright. So thank you. Peter.
Thanks, Christine and Nick. What an absolute honor to close the inaugural Bob Bartlett Legacy meeting. I am speaking basically on behalf of Dr. Brody, who's not joining us this morning because he's otherwise committed, but certainly sent his appreciation for the quality of the talks and the international participation across the globe. I want to specifically thank Nick Barrett, who brought this forward as a construct at the end of last year, and it was immediately taken up like groundswell. This is clearly the appropriate way to be engaging the international community in appreciation and in celebration of the work that was started by Bob Bartlett and his colleagues all those years ago, and that's really been continued by the entire community. If you've enjoyed watching the talks, come on in. If you've enjoyed contributing through speaking or moderating, come and be a part of the community. There's so much work to do in this space, and doing it together is the best form of collaboration. We, as part of the ELSO team, are happy to coordinate anybody's interest and investment in this space. And building together is the best way to continue to honour the Dr. Bartlett legacy. I'm really looking forward to doing this again sometime. I could say that now that we've survived the 24 hours, and look forward to seeing you all at global meetings as we continue through the year. So thank you, and this is the official wrap up. We'll see you next time.
Thanks, everyone.